All right, there we go. So I'm going to start over for the recording. Welcome everybody to Hash Church season 12, episode 4. We got a pretty special episode together this Sunday. I'm not going to lie. I saw Chimera do his presentation at the Grow Up. It was very loud. It wasn't the best setting for him, and we decided together, why don't you come do this presentation, expand it a little bit on Hash Church, and then we'll have a lovely conversation after about it. So without further ado, I would like to shout out, of course, my sponsors, because we love the sponsors. They've been so good to us. We're going to start with my good friends over at the Press Club. Today's episode of Hash Church is proudly supported by the Press Club, California brothers obsessed with hash and everything solventless, just like us. Their journey didn't start as a business, it started as a family back in 2014 when a loved one wasn't finding relief through conventional medicine. They turned back to the plant they'd respected their whole lives. They knew cannabis had medicinal power, but they needed something cleaner. That search led them to solventless, using nothing but water, heat, and pressure to let the plant express itself honestly. That light bulb moment turned into washing, pressing, experimenting, and eventually realizing how hard it was to find truly high-quality rosin bags and solventless tools that were made in America and still affordable. So they did what real hash heads do. They built the solution themselves. The Press Club stands for clean process, conscious intentions, and tools made by people who actually live this culture, not corporations chasing trends. We're grateful to have the Press Club supporting Hash Church and the wider solventless community, family-run, purpose-driven, and rooted in why hash matters in the first place. Much love to the Press Club, and really thank you guys for supporting Hash Church. Of course, you can reach those guys at www.thepressclub.co. Use the code HashChurch10 or Hash Church for a 10% discount on their wares. And, go give them a shout out because they've been supporting us so well. We want to turn around and support them as well. We also want to talk about Puffco, who have been absolute incredible sponsors. They just signed up for another year at Hash Church. At Hash Church, we talk a lot about ritual respect for the plant and elevating the experience, and that's exactly why we're proud to be supported by Puffco. Puffco continues to set the standard for modern consumption with tools built for people who truly care about flavor, temperature, and intentional use. From the Puffco Peak Pro with the 3D XL bowl delivering consistent heat, bigger hits, and unmatched terp expression, to the Proxy, redefining modular ritual-based consumption, and the Pivot, bringing true Puffco performance into a compact, everyday format. These aren't gadgets. They're purpose-built tools for hash and solventless. We're genuinely grateful for Puffco's continued support of Hash Church, our guests, and our community. Their belief in education, culture, and quality helps us keep these conversations alive. You can support the brands that support the culture by visiting www.puffco.com, explore their lineup, and experience what intentional design really feels like. Puffco, elevate the ritual. All right, well, we got to talk about, last but certainly not least, we wouldn't be here without the pioneers who helped define solventless extraction, and bubble bags are a cornerstone of that history. Bubble bags helped introduce the world to ice water extraction, giving growers and hash makers a simple, effective way to separate resin using nothing but water, ice, and agitation. While a little bit of herb will help as well. Decades later, our bags remain a trusted tool for people who care about purity, process, and tradition. From home runs to professional washes, bubble bags continue to support true solventless craft, allowing the plant to speak for itself. We're proud to be supported by Bubble Bags and grateful for their ongoing support of Hash Church, our guests, and the global hash community. Their commitment to quality and accessibility has empowered generations of hash makers. You can support the brand that built the culture by visiting www.bubblebag.com and explore their extraction bags and accessories and keep the solventless tradition alive. Bubble Bags, water, ice, hash. Awesome. Thanks, everybody. We're just going to continue on here now. Let me see if I can't. Oh, there we go. I'm excited for today. I'm not going to lie. I don't see Ryan in the room yet, but I will send him over the link right away because, he's got a nice little presentation he's going to be doing. Oh, he's got Zoom issues, so he's rebooting. No problem. I can chat a little bit because I just got finished with the Grow Up conference. Thanks to Randy Rowe for putting on such an incredible event. Very much focused on the legacy, although there are tons of legal companies. The entire vibe of the show is very, very legacy oriented. We shared our booth with Valen Veras. So Ruben and Alex Goldberg were at our booth. Thank you to Katie and to Drake, who helped run the booth. And of course, huge shout out to Sambo Creek Filtration, whose booth it actually was. We had a beautiful plasmatic sifter at the booth during the show. People loved it. Tons of people came up and saw it. We had a couple of people interested here locally who may want to buy the machine that I had. And it was just a really great show. Lots of wonderful people, lots of beautiful resin. Shout out to the Sauce Boys who hooked me up with this little ditty. I can't even remember the cultivar at this point in time, but it was a beauty. It was written on the top here, and now it's gone. Gas something. I can't really remember, but thank you to Hugo and Jordan. We look forward to having those guys as sponsors again this year. We spoke about it briefly, and we're going to have another conversation here soon. I also want to shout out my boys, Malik and George at Rocky Road, Aurora, and Thornton in Denver, Colorado. I finally got to meet these guys. These guys are massive supporters of what we've got going on in Denver. I'll put up a little picture of me and Malik the last time I was at the Aurora store. How cool is it that these guys put a full-size wrap of the Bubble Man logo right in front of their store? If you're in Denver, if you're in Colorado, head on down to Rocky Road, Aurora, or Thornton. Go say hi to George. Go say hi to Malik. They've got an incredible team over there. They were just oh so generous and such great folks to hang out with. Really big shout out to Rocky Road. I know we're going to have an incredible relationship with the Bubble Man brand. These guys are selling our hash hits. I believe they just ordered our live hash rosin pens. And they may or may not have our bubble and our live hash rosin, but I know they're excited for the pure hash joints and some of the other novel products that are headed down the queue for them. So yeah, if you can, go say hi to them. Tell them Bubble Man sent you. When I was there, he was also very generous, hooked me up with a hoodie, bought me a couple of Keef Colas, and got me some of the Tikal rosin. I had not tried Redman's brand Tikal. I'd seen he had a lot of flour for sale in these incredible boxes that he'd been giving all his hip hop friends. So shout out to that whole crew, and shout out to Tikal and Redman. I may be or may not be working with a group in New York who is very, very closely related to those guys. So we may see some interactions on those two brands down the line. Welcome, Mr. Ryan Lee, who just stepped off camera. As I said, welcome. That's always the best. It's always nice when your waitress or waiter walks up to you and asks you how the food is right after you've jammed a shovelful into your gullet. Kind of the same thing as welcoming Ryan right after he steps away from his chair and walks off camera. So I'm a classy guy. I'm on the ball. It's Sunday morning. I'm exhausted. How you doing, Ryan?
I figured it would be a good time to get a drink while you were doing the introductions.
Apparently not.
That is funny. The waiter thing. It's inevitable how they do that. But
I swear, I've never worked in I've never been a waiter before, but it has to be somewhat on purpose sometimes. Like it
you
must be
think
a
so,
rolling
yeah.
joke.
Yeah, well, I guess you don't have the opportunity to really say it sucks, right? Like bring it back, bring something back.
No, it's happened to me 1,000 times. Excited, man. Stoked to see this presentation. I think you did an absolutely stellar job at Grow Up. Grow Up. I always call it Grow Up. I wonder why. But I think a quieter environment, a lighter environment, and I know that you've gone through it a little bit more and sort of dug in a little bit deeper, maybe expanded a little bit. I'm super excited, man. I'm not going to lie.
Yeah, I mean, and it's such a huge topic, right? Like breeding cannabis is such a massive topic. So to like cover it all in an hour is, frankly, it's insane, right? So this should be really seen as like a review, not necessarily a review, but like kind of an overarching quick first path of all the important things. And then, like we could honestly spend an hour on pretty much every slide and the details of what every slide means, but we're obviously not going to do that
We've got four
today.
hours, so we can do what we want. I loved your idea of however long we do today and the discussion about it is to bring this up kind of as a breeder's corner every episode of Hash Church, where we do a little bit of a, just keeping everyone. What I loved about the grow up is you really walked us through the steps of kind of understanding, and then the conversation could begin. So I'm hoping that everyone gets that out of this today.
Yeah, well, the first half is kind of like it has to be based in theory, right? And that's kind of the stuff that I want to really push home. That I think a lot of the breeders in the community miss this part. And it's really not good to miss this part because this is like the biological reality that underpins everything we're doing, right? Like there's no arguing with chromosomes and meiosis and mitosis and cell division.
Mm.
So the realities that govern those processes and the effect that that has on breeding is really important to understand. So that when you do see traits, you can try to figure out how they're inherited and how to, like, once you know how they're inherited, then you can breed those traits into other lines,
This has
right?
always been the thing that is kind of beyond me. I've never quite, I mean, I understand you're looking for traits. You have to understand how those traits move through the progeny, but I just don't, I've never understood like, what did Dave do? Like he just went into the grow room and he just bred this particular thing into plants. Like it's very, very interesting. I'm looking forward to listening to it again today because it was a little bit loud and I was very focused on holding your phone and recording it. Today,
Yeah,
I'm
that was a
going to
burden.
relax with some dabs and just listen.
Cool. Well, I got some good hash too at the show, so I've had a couple of dabs this morning, as you can probably tell from my eyes. Morning,
Excellent.
Colin. So let me see if I can see what the sharing thing here. Here we
Perfect
go.
timing, Colin.
Good. The snow is going nuts over here on the East Coast, so I had some preparation, so pardon my tardiness.
You got your Jenny all hooked up or what?
Got it all hooked up, man. I got the firewood chopped. Yeah,
Do
you
you know
know.
what Leo Sue said about firewood?
No,
He who
enlighten me.
chops his own wood heats himself twice.
Ooh,
There
that's a
you
good
go.
one.
It's true. Do you have to do something with the sharing, Marcus? It says I'm paused here.
Well, it says Ryan has started screen sharing. Double click to enter full screen mode. No, I mean I accepted it when you asked.
What do you see?
I just see a black screen that says Ryan has started screen sharing. Double
Yeah,
click.
that's what I see too, and I don't have any black screens, which is the problem.
All right, well let's try it again. That's a
All right.
weird. We've never had that problem. I'll make sure I go into my share button, share settings, and I'll make sure that everyone can share because that would really suck if
You're
this
still
didn't.
having the black screen. It's funny.
Yeah,
I've
I'm still having the
never
black
had
screen.
this
You've been
problem.
having no, but it's like today we're trying to do something. This is how Hash Church works, dude. Share
Dave,
Truth.
selected
Dave's
app.
having a blast with us somewhere.
Yeah, he's messing with us, dude.
Yeah.
Share screen. I've got recording going. I don't know. This should be able to work. We need it. We need your slides. So maybe if you go out and come back,
Yeah,
Try that.
let me just try one more time here. No, it's still not doing it. Jesus.
it's really
All
So
right,
weird.
I'll be
Ryan
back
has
real
entered
quick.
screen sharing. Yeah, no, it didn't work. All right, well, come back. We're gonna get this going. Oh man, Dango. Had a pretty wild event the last 4 days, dude. Grow up
how was it?
was wild and it was good, man. I had those Valenveras guys at my booth. They're just the most funnest people on the planet, basically. We had
Nice.
Sambo Creek filtration, so we had like the big static filtration unit. People were like, "What is that? A popcorn maker?" I was like, "Yeah, kinda,
Sorta.
sorta,
Sorta. Just
sorta,
different types of corn.
sorta, kinda." All right, fingers
D-
crossed that this just works.
Let's see it, man. You
No,
can
it's
also,
doing it.
Ryan, you can send, maybe try sending me the link and I can maybe download it and then I'll drive
Yeah,
and
you
you can
don't
talk.
even. Yeah, that's a good call. It is. You have the link already. It's that first link in the Breeding Church discussion.
Oh, okay, there we go.
Um, my phone is not on me and that's
Okay,
in a different,
let me just give me
if you
one. Let
don't
me try
mind,
one more thing. Okay.
Reading
Oh,
church. So what? I have it then?
yeah, you have it
part
as
of my,
well.
yeah, part of my scramble this morning was
Okay, that works.
Yeah, that
yeah.
does work.
Okay, so it was just the thing.
Yay! What was it? What was it?
It didn't want to select the one window, and then when I put it on the whole desktop, it worked.
Okay, well, let's just make sure I want to hope that it's going to be. We're always about 30 seconds behind on YouTube, but I want to make sure that we can see this. So before you start, there it is. Beautiful. It's on. You're on. We're on. I think it's time for a dab.
I was just about you just read my mind, dude. Come on. Read
And
my mind.
Colin, you kind of know a lot of this stuff, so if you think that I'm like breezing past something that should be focused on a little bit more, kind of, or do you think I'm going over it too quick, just stop me and we'll clear it up, okay?
Yeah, no doubt, man. Yeah, for sure. Well, we can slow down. I think it'd be great to throw some questions to the audience and um to just take temperatures on the concepts and make sure that everyone understands. Just a thought.
Yeah,
Yeah,
as questions come up, I could get Bingo to copy and paste them into a note. I don't know if that's too difficult for you, Bingo, but if you can, that way questions aren't necessarily interrupting the flow of the beginning of the conversation because it really is a presentation, but we can go back and not miss those questions.
yeah. Questions should be answered as we go along. But yeah, like if we can kind of hold most of them till the end, unless you like again, Colin, unless you think I'm blasting past something we should focus on a little more.
Sure. Okay. Fair.
Okay.
Yeah.
All
Right
right.
on.
So this was suggested to me by Jodry to present, Kevin Jodry, and they wanted something that kind of in like wrapped up a breeding program so people understood the challenges that you face and also like maybe the hurdles that you overcome to get there. And my real thought about that is that like plant breeding is not really hard. It's just figuring out, make sure you have the resources to do it, the time and the space, right? Because it really is about space. So, and how much space you're going to dedicate to it, because it does take growing a lot of plants. So we'll start with this slide here. This guy here, David Brown, is like a Canadian. He's someone involved in the industry. He's worked for Health Canada, kind of developing the nursery licenses. And he was joking around on Twitter. Who knows how much he was joking, but it was kind of a shot at plant breeders, right? That all we do is we make plants fuck, and obviously, breeding cannabis can be so much more than that. I think the general consensus is that in the industry, it's not really being done very seriously. So to kind of get a finger on how true that is, the standard approach to cannabis breeding is you got your sherbet strain and your gelato strain, and so you cross those together, and then you end up with sherblato, right? And a lot of kind of the breeding thought process doesn't really go too much deeper than that. But what is sherblato? And if you ask that question, you'll get these kind of responses like, "Trust me, bro, it's fire." There's a lot of like unmeasurable words that are kind of descripting words, but not too accurate. There's no way to really measure them. So you get like your dank, woody, herbal, citrusy, earthy, cushy, and of course every line has the best phenos ever. And that's like really like this kind of hit or miss approach where you're just mashing things together and screening the offspring. And people call that pheno hunting, but I kind of refer to it as pheno stumbling because it's such a random thing. You're really like hoping and praying that you're going to find something out of this pack, right? And on a maybe a larger scale, it's one thing if you're doing that in your closet, but if you're really on a scale where you're working in like a legal regulated market, you have to be hitting like key THC numbers because the way that the market is kind of skewed is the buyer is so THC focused that the distributors will only buy stuff that's like 25+%. Things that are in the 10 to 15% cannabinoid range are just like they have no market appeal, or no, at least to the distributors, and so a lot of that stuff ends up getting left off the marketplace. When we've seen some pretty famous victims to that, like Sour D, for example, that's like a 15 to 18% plant. Blueberry is obviously like low teens. So like a lot of these things that maybe people would like, they just don't end up getting grown because everybody's so focused on high THC content. And that's like one thing that's not the easiest thing to find, right? So anyway, the point is, growing seeds and hoping is not how we create the IP that supports this now multi-billion-dollar industry. And so we can do better for the breeding. And this is, the next question is, how do we do that? So So we come to our famous friend here, Gregor Mendel. He's the one with the M on top of his head. And he is what we now know as the father of genetics. He was a monk in this monastery in the Czech Republic. Back then it was part of the Austro-Hungarian Empire, so he's always referred to as Austrian. But you can go to this monastery still today. And he was hanging out and they're doing their chanting and all the things the monks do. But obviously back then there was no internet, there was no, books on how to understand how inheritance happened in plants. And these guys are just bros hanging out, growing a lot of their own food because they don't have, back then there was not these massive food production systems that we had now. So the monastery would grow a lot of their own food to feed, their kitchens. And so he's checking out all these pea plants. He's got all these varieties of peas. And he starts noticing that some of the peas have red flowers. Some of them have white flowers. Some of them have round peas. Some of them are wrinkled. Some of the peas are green. Some of the peas are yellow. And he's noticing that they all have these traits, okay? And the way that pea breeds is every flower is, it's what we call a perfect flower. It has both sexes on the same flower, which is obviously different from cannabis, which has males and females. So peas undergo a natural amount of selfing, which cannabis plants don't really undergo. And because of that, the traits that they possess are relatively true breeding, or they become true breeding over time. And so the red flower plants always produce red flowers, and the white flowered plants produce white flowers. And, the traits that he's seeing in all these plants is quite consistent. And he's curious about how that happens, or why that happens. And so he starts intentionally, you can do something on a flower called emasculate it. You can go in and you take off the pollen-producing parts of the flower. And then when you use the pollen-producing parts of another flower, of a different variety, you can force an F1 cross between these two types. And so he starts doing all these experiments. Now, keep in mind, it's he's doing this work starting in 1856, ending in 1863. Just for context of the time period, Origin of Species is from Charles Darwin is published in 1859. And the Civil War is 61 to 65 kind of thing. So, it was a fair while ago. And they didn't really have an understanding of what a species was, how breeding worked. And so he's trying to crack this puzzle. And so he's following all these different traits, but only when he starts doing the math at the end and trying to figure it out, only 7 of them really kept together. Now you have to keep in mind, to do this work, you're keeping immaculate records of the traits that the plants have, each plant. You're taking lots of notes. You're isolating the seeds and keeping them from other people coming and trying to use them in the kitchen, or your nephews coming by or whatever, messing with your seed collection. Everything has to be done really carefully and collected very separately. And so he figured out a lot of the stuff, published the work in 1866, and nobody even really knew about it until the 1900s, when a few researchers independently rediscovered it by looking through the, back then there was no internet to be sharing scientific papers. So people were going through libraries and old books to try to find things about inheritance. And in doing so, they finally eventually recognized that this work he had done so long ago was really quite interesting on how it showed a likely mechanism for inheritance of plants. So we're going to go through that. So these were the 7 traits that he followed. the really visible ones are these kind of top three on the left. We've got the round seeds and the wrinkled seeds. We've got your yellow seeds and your green seeds. Red or pinkish flowers or white flowers. Obviously, like the length of the stem, color of the pods, and then even like shape of the pod were some of the other traits here he was looking at. We're really going to focus on these top three on the left here. So, Mendel realized that he had, these pea plants with red flowers and these pea plants with white flowers. And when he bred those white flowers to themselves, all the seeds came up with white flowers. Same for the red. If he bred the red to themselves, all the plants came up red. And so he forced a cross by cutting off the pollen-bearing structures in the flower on one of the varieties and cross-pollinated them. And he noticed by, very carefully collecting the seeds and planting them again the next year that all the seeds were red and the white flowers had disappeared. If he made an F2 of those seeds, if he crossed those seeds between themselves, the resulting seed the next year were I'll show you here. So this is the next year's cross. And the ratio that appears is this 3-to-1. So whereas that white flower that only came, it went into half the genetics, now it's underrepresented. It's only present in one of the flowers. And so he decided to use this terminology of dominant and recessive traits. And he decides that in the first F1, in the F1 cross, which is this first hybrid generation here, that the red color dominates the white color. And then what he names as the recessive is the recessive is obviously white in this case, and that's what he calls recessive, just because of the way that it behaves in the cross. And so he comes up with this concept of alleles, and alleles are like these what he calls heritable factors, because he doesn't know anything about chromosomes. We've obviously since come to learn about chromosomes and understand that these traits or alleles are actually just different versions of a gene. So on every chromosome, you have what's called a locus. A locus is like a coordinate or like a location. And so a specific gene, we can call it THC acid synthase, is at a specific spot on one of the chromosomes. And you can have different alleles for that gene. So you can have a gene that, for example, a gene that is functional or a gene that is non-functional. That's one set of alleles. So an allele just means different versions of a gene at the same spot on the chromosome. And these different alleles in combinations with each other lead to different phenotypes. And that's driven by what we call a genotype. And so the red genotypes, or the red phenotype flowers, are governed by this dominant condition, which we call big R. The recessive condition, we give the label little r. So the true breeding red flower, this P generation, we give those the designation RR, and the F1 generation would be RR. In this case, they both lead to the same phenotype, and it's the little r genotype that gives the white phenotype. Okay? And again, in the F2, we always see this typical 3-to-1 pattern of dominant to recessives. Okay, so just again to sum it up, alleles are versions of a gene at a genetic locus, which is a location on a chromosome. We have two sets of chromosomes in every cell. One set of chromosomes you inherited from your mother, one set of chromosomes you inherited from your father. It's the same in all diploid species, so mammals and most plants are diploid. I think all plants are diploid, actually. Well, not all of them, but and homozygous just means we have two copies of the same gene. The zygote is the first cell that's formed when either a sperm cell meets up with an egg cell or a pollen grain meets up with an ovum in a flower. And so homozygous just means same in the zygote. Heterozygous means different in the zygote. And we represent those genetic conditions by big R, little r, are homozygous. And then the heterozygous kind of hybrid condition gets the big R, little r designation. Okay, so he starts looking at are traits inherited in combinations with other traits. Like if we have round yellow peas and we have wrinkled green peas, what happens when we cross them together? So both of these lines, round yellow and wrinkled green, are true breeding. If we plant them, 100% of the seeds come back matching the same phenotype, either round yellow or wrinkled green. But when we cross those two plants together, all of the offspring are round and yellow. And so this is our what we call a Punnett square. A Punnett square is just to look at the different genotypes and what happens when we cross them together. And so we have, this is just another way of saying the big R, big Y is one of the, that's the only type of gamete that can be produced by the round yellow seed. It has the two dominant genes. And then the wrinkled green pea is this one on the left side, and that's denoted by little r, little y, because both of those conditions are recessive, okay? He's already figured that out. So here's the Punnett square showing that all of the options are, all the possible combinations from this cross are round and yellow. But he makes the F2 cross, he crosses that first generation within themselves, and all of a sudden we have two new classes. We get 9 of the original round yellow, we get 1 of the original green wrinkled, and now we have these two new combinations of round and green and wrinkled and yellow, okay? And those things didn't exist before. And you can see that our Punnett square proves this math out. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 of the yellow round. We have 3 of the green round. We have 3 of the yellow wrinkled and 1 of the green wrinkled. And that is our 9 to 3 to 3 to 1 ratio. That's a very classic, it's a classic number of categories that we see come out when we make these dihybrid crosses in every plant. So this is kind of just the math behind why that happens, okay? And again, we're kind of saying the same thing. An F2 of a dihybrid or 2 gene cross has the genotype segregation pattern of 9 to 3 to 3 to 1. I should actually say phenotype segregation pattern. I'm sorry. Now we can look at those two independent traits together. The green pea and the wrinkled pea. They're both recessive. The probability of each of those traits is 1 in 4. And we know from basic math that the probability of 2 independent events occurring simultaneously is the result of their respective probabilities multiplied. So we have 1/4 for one mutant phenotype times 1/4 for the other mutant phenotype. To get both of those back, we only get 1 in every 16 plants, and that corresponds to this 1 in our 9 to 3 to 3 to 1 number. And so he thinks that's interesting because that plant came in, that green wrinkled, the green wrinkled pea plant at the beginning of his experiment came in with half the genetic material, but the phenotype only came back in 1 of every 16 plants. So he thinks that's kind of neat, and he's keeping really, really good notes, and he sees that this pattern is happening in all of these different experiments that he's doing across all these traits that I showed you before that he's following: leaf, pod shape, pod color, leaf flower color, etc. And so he comes up with these 3 laws, which we learn down the road that they have. They don't quite explain the whole story, but they explain a lot of the story. One of them is what he calls the law of dominance, and that's that these dominant traits, like the red flower color, they mask the recessive traits for at least one generation, or for one generation. We have the law of independent assortment, which says that these sets of genes related to each, like green, for green colored peas and wrinkled peas, are separate And he says that the way he puts it is that they assort independently, which means that we can have this recombining of traits that we thought they come together in sets, and we can actually uncouple that combination and create new combinations. And then he has his law of segregation, and that's these parental alleles that are these alleles that are inherited from the parents. They don't have to come together, and they can separate during the formation of pollen grains or the egg in the flower. Okay. This is an example of something that Mendel didn't describe, but we've since learned that is true. It's a different type of interaction that's kind of halfway between dominance recessive. Sometimes you cross a red flower to a white flower and you end up with a pink flower. And in the F1, all the flowers are pink if red and white are both true breeding. And then when we make F2s out of that cross, we end up with 3 classes. We got 1 red, 2 pink, and 1 white. And so that's kind of a modified version of our 3-to-1, right? Like you can put the colored class together and that 1 plus 2 equals 3, and then we have our white. And so we explain this by what we call codominance, where both traits have an impact on the phenotype of the offspring. Okay, and that codominance in the F2, when we see codominance, we always get that 1 to 2-to-1 pattern in the F2 where we recover the two parental types out of the F1. Okay, so we're getting through all the kind of the basic biology stuff. We will soon get to the cannabis specific stuff. Okay, so cannabis breeders really must understand a series of things that we're going to go over. One of them is the difference between inbreeding and outcrossing and the limitations of each of those methods. Outcrosses increase the genetic diversity within the population or within the family that you're creating by making the cross, but we lose uniformity. So that's not always a good thing, right? If we're trying to follow a trait or we're trying to fix a trait that we like in a plant into a seed line, we don't really want to increase the genetic diversity because it makes it more likely that we're going to lose that trait to a dominance or recessive relationship. We also have to understand that inbreeding decreases genetic diversity and increases uniformity over generations, which is a good thing. But if we inbreed too tightly, we can run into what we call inbreeding depression, which is essentially the genetic weaknesses in any line also become reinforced, and the plants can suffer in terms of growth or yield or fertility or terpene production, right? We can actually start doing harm by inbreeding too much. So it's something you have to pay attention to and account for as you're making your plans, your breeding plans. Okay, so you have to have an understanding of these different types of breeding. We have, say you've got your sherbet and your gelato. Let's call those your parentals. The F1 cross is the first hybrid between those two plants. We've now created a family from those two parents. Within, humans, it's not really so done, but in plants and other animals, we breed brother to sister when we're trying to fix traits. And in plants, it doesn't actually really have a genetic consequence unless we do it too much. If we make that first cross and then we stay breeding within the family, that's what we call a filial cross or sibling crosses. So those kind of crosses get designated, like we name the populations F1, F2, F3, okay? A self-cross is when we take one of these F1 individuals, usually an F1 individual, and we pollinate it with pollen that's chemically induced in the plant by something like STS or ethylene. When you produce these silver compounds onto a female cannabis plant, it blocks one of the hormone pathways called ethylene, and that results in the formation of male flowers on a genetically female plant. And so when you cross that male, that chemically induced male of a clone back to a female plant, you get what we call a self population, okay, or a self S1 generation. This is a population that is derived from one individual. So we're able to see what happens when we start recombining and segregating the traits of found in one individual, and that can actually be really helpful, because we've limited the population to only 2 chromosomes. When you're breeding from within a filial population, the F1 generation, they all have 1 chromosome from mom and 1 chromosome from dad. But the brothers and sisters within that populations, they have different chromosomes from mom and dad, right? And so, even mom and dad each had different chromosomes. They had one from each of their, of the grandmother on the paternal side and the grandfather on the paternal side. And so these chromosomes in those grandparental forms end up in the filial generations. And so when you're crossing brother to sister, you're not necessarily limited to 2 genes, right? But when you do self crosses, you're limited to 2 alleles or 2 chromosomes, and that is really helpful in analysis because it allows us to understand more easily what's happening in how a trait is inherited. And then, of course, we've got the intergenerational back crosses, which are highly popular in cannabis. If you follow my Twitter, or my Instagram, you see me rail on double back crosses quite often with memes. Anyhow, it's important to understand what each of these type crosses are and what the implications are for that type of breeding. Okay, so this is a new thing we didn't talk about at the conference. This is just a basic diagram of a plant cell. There's some basic biology stuff that happens when cells grow and divide. All tissues, as we see them dividing in the plant leaf and growing out of the meristem, that's the result of cell division called mitosis. And so I just thought it would be important to kind of give you guys a little bit of a look at, some of the basic parts of a cell. You've got your cell wall, which is quite rigid, the cell membrane. We've got the mitochondria, these little pink guys, and the green ones are the chloroplasts. The chloroplast is obviously the part that gives the plant its green color, but that also harvests the sunlight and the energy for the plant. And then we have the nucleus, and the chromosomes are within the nucleus. Okay, so that's just a little basic plant physiology there. We also have to understand the concept of haploid cells and diploid cells. Haploid cells, so when you think about all the cells in your own body, we have what are called somatic cells. The somatic cells are the cells of your body, and then we have gametic cells, or the gametes. And those are the cells that are produced in the testes or the ovaries. And those types of cells are different than the rest of the cells in your body, because the ones in your body have 2 sets of 23 chromosomes. So we say 23 chromosome pairs. But the sex cells, or the gametic cells, they only have 1 set of chromosomes. Okay? So what happens is when those cells are made, your body packages up either or of the chromosomes that are inherited from your parent. And so you kind of give this shuffling of the chromosomes that you inherited from your parent, from your mom and your dad, and your offspring will have a combination of those traits. But in the rest of the cells of your body, those cells or those chromosomes are still separate. And so it's important to understand that these cells have 1 set of chromosomes, because when they come back together in the pollen grain and the egg within the flower, that reestablishes what we call the diploid condition, or the 2 sets of chromosomes level. Okay, so I hope that's clear. This is a really important part of breeding, so we're going to go over it a couple times. So again, we call this ploidy. Okay, so ploidy really refers to the number of genomes or full chromosome sets within a given cell. Okay, pollen grains, sperm, the ova in the flower, and the eggs in the mammalian cell, they are all haploid. They have 1 N, and that 1 N just means 1 full set of chromosomes. When those when the sperm and the egg come together, or the pollen and the ovum come together, we get what's called a zygote. Okay, and that new cell reestablishes the set of the chromosome set to 2n. Okay. You probably heard of polyploid in the context of like seedless banana or watermelon, and they're now starting to make polyploid cannabis seed, where we create plants that have 3 sets of chromosomes. And we do that by crossing a 2n plant and a 4n plant, and we end up with a 3n plant. Now, the 3n, because it's an odd number, that induces this kind of chromosome instability, and that leads to seeds that aren't really formed properly. Like if you look at a banana, you see these little black dots in the middle of the seeds. Those are kind of like seed abortions. They're seeds that didn't like form properly. In watermelon, they're the little white seeds that you see in the watermelon, in the seedless watermelon. David tried to do this. David Watson, Sam Skunkman, obviously, who is, someone we all know. He wanted to do this with cannabis seeds years ago, and he had a lab in Hungary make for him triploid cannabis seeds. They doubled the chromosome number and then crossed that back to a 2n plant. And he found that, like the watermelon, cannabis still set seed, and it had these little, what he called whiteys. And he decided that those were useless. The idea was to be able to make a plant that was sterile, so you could grow drug cannabis in the middle of a hemp field, for example. One might do something like that. But the plants produced these like half viable seeds, and so they were they didn't end up being the success that he had thought that they might be. Just for interest, wheat is hexaploid. It has 6 sets of chromosome, and strawberry has 8 sets of chromosomes. So we've been playing with ploidy with plants for quite some time.
So Ryan, just like a slowdown for
Yeah,
here. Like
of
I
course.
think this is important. I think to distill a little bit because what that does, what Ryan just went over, the haploid, diploid, polyoid. I think the to distill it down, I think is important here because what that does is really increase genetic diversity and buffering where it gives it extra chromosomes. So like, let's say you fed it wrong it can deal with those types of stressors and tolerates things in an environment where other genetics that are, let's say, a bag seed, which I think you can speak to, are not as strong or nor really at the same caliber of genetic diversity. So you're able to have a stronger garden just by design. Like, and I think that's really important for people to really realize. So if you can just talk about like what it increases biomass and it's better sterile seeds if you're breeding doesn't allow the plant to in some cases be pollinated, right? Some people like it for those reasons because they're growing, let's say, outside. So the use case, I think, is so important because what you just covered really is the kind of a roadmap to creating more diversity, right?
Yeah, it's a different. Yeah, so polyploid, the real thing that it was sold as is it's going to make plants sterile. And it just didn't make them fully sterile,
Yeah.
right? And so it kind of failed in that regard. But polyploids, because they have extra sets of chromosomes, they do have weird traits. Like some of the leaves are bigger, the stems are bigger, the kind of the breathing holes on the bottom of the leaf are bigger. And reportedly, they might even produce more oil. So there's reasons why you'd want to use those plants. Again, especially if you're growing in an area where they're growing a lot of hemp, and you're trying to grow flower for extract, for example, or whether it's terpene extract or cannabinoid extract, when a plant gets seeded, the plant puts all this energy into the development of the seed, right? Because that's the primary focus of the plant, is it's trying to make seeds for next year's generation. That's what it's evolved to do. And so we're kind of doing something by not letting these plants get pollinated that kind of hijacks them for our own purpose, right? And when they're not making seeds, they produce a lot more oil on the plant, or more, stronger glandular trichomes. And so in pollinating these plants, you're kind of taking the energy away from that energy that's going into the production of essential oils or scent or cannabinoids. So people started playing around with ploidy to see if it would work, and it has worked to a degree. Arguably, some of these plants produce more terpenes. With 3 sets of chromosomes, there's potentially more opportunity for the way that the terpene synthases work together, but I think that has to be proven. But it's something to keep in mind as we go along. I don't really want to focus on the polyploid stuff too much. The important part for this next section is focusing on the haploid cells and the 2N cells, because we're going to go through meiosis and mitosis. Okay. So, we talked about humans have 23 sets of chromosome pairs. Cannabis has 10 sets of chromosome pairs. One set of those 10, we call the 10th pair, we call the sex chromosomes, the X and Y. Males have X and Y, females have X and X. The 9 other pairs of chromosomes we call the autosomes. And just, some basic genetic biology, chromosomes are obviously the larger units that contain the gene subunits. If the genes are on the same, if the genes are the same on each chromosome pair, we call those homozygous for a locus, or true breeding, and that's, they can be said they breed true. Obviously, if the chromosomes have different alleles, like we talked about, different genes, we call the plant heterozygous. Okay, so this drawing is essentially meant to show, it's a model for how cells grow and divide. The blue chromosome will call from mother, the red chromosome will call from father, but really, like, this 1 and 1 actually represents 10 chromosomes, but we've modeled it out here with 1 chromosome. And so what happens is, the plants, the cells occur in their natural diploid state, but when they go to multiply, when the cell goes to divide, the DNA doubles, and so we get an exact copy of each chromosome set in the each chromosome in the cell. And so we go from 10 chromosomes to 20. We go from 10 chromosomes pairs here to 2 sets of 10 chromosome pairs. And then those chromosomes separate to opposite ends of the cell, and the cell pinches off, and we get 2 new diploid cells at the end. So we start with a diploid cell, we get 2 diploid cells at the end of that process. This is a longer explanation of how that happens. In interphase, the cells are just kind of like hanging out. The chromosomes are in the nucleus in the cell, and the DNA is kind of all uncoiled because the genes are being read and transcribed into proteins. But before the cell goes to divide, we have those chromosomes form back into their structural units. And through prometaphase and metaphase, the chromosomes they dissolve out, the nuclear envelope kind of dissolves, and these chromosomes all line up at the center of the cell in what we call the metaphase plate. And you get these like spindle fibers. You can see these little lines that are going back and forth between the 2 poles of the cell. They kind of look like lines of latitude on a map. You can kind of think of those things as like zip lines for chromosomes. And so the chromosomes at the centromere, the center part of the chromosome, they attach onto these spindle fibers, and they kind of zip line toward the poles of the cell ends. And that process is when so we have the chromosome doubling, the cells all line up along the middle, and then they split back in half, and we end up with 2 daughter cells that are both diploid again. Okay? So this is a process we call mitosis. And this is what happens every time a cell in a root doubles. When the roots are growing, the cells are actively dividing on the tip. This is what's happening. Same with the Same thing is happening in the meristem on every shoot leaf junction, right? Or any auxiliary bud where we have new growth happening. This is happening a lot. Cells are dividing, they're doubling their chromosomes, they're dividing into new cells, and we end up with more new cells. So in the male flower and in the female flower, we kind of have this double, you can kind of think of it like extended mitosis. But it has a second step, and that second step results in, well, I'll go through it. The first thing starts again, we have the same thing that happened in mitosis. More or less all the same steps happen. You get the doubling of chromosomes, they line up along the center of the plate, they split in half, and we get one cell becomes two cells, okay? Which we'll represent by these two here. But what happens on the second division, there's no doubling of chromosomes. We just have separation of chromosomes. And we go from a diploid cell, which is 2N, two sets of chromosomes, and back down to the one chromosome cell type. Now, this is really important in any breeding, because this is where the reshuffling of traits happen. This is where, any given cell is ending up with the chromosome from the mother or the chromosome from the father, right? And just because you get chromosome number 1 came from the mother of the plant, chromosome number 2 might be from the father of the plant, and chromosome 3, for example, might be from the father of the plant. And so there's this like random redistribution of chromosomes in the daughter cells, in the sex cells. These become pollen grains or ovum or sperm cells or eggs in female mammals. And that kind of chromosome shuffling that happens right here is really the most important thing that is happening in plant breeding, because that random recombination is where we can now go into the plant and select for what we call recombinant plants, where you have, or recombinant traits, where you have specific combinations of traits from the mother and the father lines. Okay? So we'll keep coming back to meiosis probably over and over again in the coming episodes of the show. Okay, so now we're kind of through with the biology. We can start focusing on the cannabis stuff. If you're starting a breeding program, you have to have a why. You have to have a reason or a goal. And you really need to define your goal before you start, right? Because we're not just crossing plants to see what happens. Sure, that's fun. But if you're really making a breeding project, you have an idea of something you're trying to achieve. You're trying to, make a plant powdery mildew resistant, or give a seed line a specific scent profile. So to do that, we want to work with or find plants that contain the traits that we require to make our, it's kind of like, think of it like putting together a dish. We need to find all the ingredients for the dish. We're always going to select plants that approach the goal, but any plants that don't get closer to what we're trying to do, we're going to throw those things away. And we call that roguing. The definition of a rogue is a dastardly fellow. And so in the context of crop, working with crops, roguing really means to remove inferior or defective plants or seedlings from a crop. Now, we're plant breeding, and it's all about statistics. And so every time we throw away a plant, we want to have recorded as many traits about it as we know, because it can affect our total outcome when we start doing analysis. We want to observe, test, and measure. We have this saying, if you don't measure it, you can't manage it. And so anything that we're trying to, any goal, a trait that we're chasing as part of our goal, we want to have a way to measure that, whether it's terpene profile or cannabinoid content or, length of the internode or, size of the flower, whether we're, taking total mass or trichome density. We always have to have a way that we're measuring that trait. And when the traits differ between the individuals, we want to count the number of individuals within every type of the trait. And we're going to keep meticulous notes like Mendel did on every plant, whether we keep it or kill it. And this is what allows us to understand the patterns of inheritance when we do the analysis after harvest. Okay? This method here, thin layer chromatography, is a really efficient and quite cheap method of chromatography, which allows us to look at cannabinoid type and profile. We use these. It kind of works in the same process that, like, if you put a paper towel in a liquid bath, kind of standing up, and the bottom of the paper towel gets wet, we see the liquid move up or kind of wick up the paper towel. And so this process is similar to that. You take like these little kind of metal aluminum strips, or sometimes they're glass or plastic plates, and they've been coated with you can kind of think of it like a paper towel-y type material. It's made out of silica. And we sit the bottom of this plate in a solvent bath here. So we've got our container, we've got a little bit of solvent right at the bottom, and when our plate sits in the bottom, the liquid moves up. And what we've done is we've taken a little plant sample and we've put it on this line. Now you can see this picture over on the right. This is kind of to show the process or give theory to the process, how it works. What they did is this green dot, when it started out, they put a big black mark of a black marker on the pen, on this thin layer chromatography plate. And the black marker dumped a bunch of ink on the plate, and then they put that ink in a solvent bath. And what happened is, as the liquid moves up, it wicks along the surface, it pushes these molecules that make up the black pigment apart. And they experience drag on the paper towels. And what happens is you see that black dot was actually made up of a whole series of colors that end up separating on the silica plate. Okay? And so when we extract cannabinoids from a plant and we put it on, we take an ethanol extract, or isopropyl extract from cannabis leaves, there's a little bit of cannabinoids in that extract. And so when we put that on a thin layer chromatography plate, for example, down here at the line, and we put it in a solvent bath, these solvents kind of push along the solvent front and they separate the cannabinoids. And so we could have, CBC or CBD or THC might be up at the top. And that's exactly what we did. And this is, we didn't invent this. This is a common technique they've been doing for quite a long time. And it allows us to separate out the cannabinoids in the mixture. And we can visualize, after we develop this plate with something called Fast Blue B. It's just a dye indicator that dyes the THC and CBD. We can see that there's different types of plants. Like this one on the left here, it has a band where there's THC, but the next plant over doesn't have the THC band, but it has the CBD band. This plant here has both bands. This plant here has only the THC band. So for very little money, we can use this really simple, low technology process that anyone can do at home to separate type 3 plants, which are the pure CBD ones, from the THC producing plants, right? And so using this process, we're able to get rid of plants that have THC or get rid of plants that have CBD. And that's really helpful to have a cheap way to do that in a breeding program. Okay, so this is work that was actually done at Heart Farm. David was making, he was trying to make these pure lines with CBD and THC for GW Pharmaceuticals. And so they were doing all sorts of experiments with crossing back and forth and learning how to keep them pure and how can they purify the THC. And he realized with his plant breeder, Etienne de Meyer, that when they crossed the pure CBD plants to the pure THC plants, that all of the F1 generation had both THC and CBD. And so they were really the first ones showing this using science. It was kind of right before the feminized seeds really busted out into kind of took over the market. And they were using STS to self these plants and kind of make F2s out of these F1 THC plant seeds. These F1 hybrids, these type 2 plants that had both THC and CBD. And what did they notice? That 25% of the plants in the offspring had CBD only, 25% of the plants had only THC, and 50% of the plants, again, had that THC CBD mixed phenotype. Now, does anyone notice anything about this? This is, of course, our Mendel's 1:2:1 ratio that we saw in codominance. And so what we're seeing here is that cannabis essentially follows Mendel's genetic model, of course, and that we can actually monitor traits and learn about their inheritance or how they create new combinations just by doing the same analysis that Mendel was doing. Okay. So now we know, we've got the type 1 plants. We know how to go from type 2 plants back to type 1 and type 2 plants, and we know how to go from type 1 and 2 plants to type 1 and type 3 plants back to type 2. So we've kind of got the switch figured out for breeding CBD in and out of different lines. And in 2013, 2014, this is kind of around the CBD craze. So everybody was like, CBD crew was around crossing CBD into every line that they could. And we're going to use this example of a plant called Renee. Actually, Renee was a very special plant. I first learned of it through Marcus. I was working in a head shop in Ottawa at the time, and Marcus walked in trying to sell some bubble bags, and he had a sample of this Renee hash, which made incredible, very melty, incredible bubble. And it had a really distinct nose profile. It was like, I described it as like a raspberry skunk. But it was really kind of bubbly and fruity and candy-like. It really had a special, unique nose to it. And I'm sure, obviously Marcus knows the plant. He could pick it out of a bag anywhere. But what happened is when we saw these CBD plants coming in, this local breeder was crossing CBD to Renee. And of course, the seed line becomes known as CBD Renee. But when you look at the plants, none of them have the special scent profile that Renee is known for. So why do people end up calling it CBD Renee? Like, Renee is really the name that we gave that plant that has like a very unique, describable funk. And, this is no different with, CBD skunk or CBD Nordal or CBD Afghan or whatever variety they're calling it these days. But really what we're seeing here is just another example of Shirbalato, right? When people are just crossing two things together and giving it a name. And it's kind of what is that name is really just more of an ethereal concept than, the way that we identify plants. I know, Colin and anyone that's experienced on this, on church, we identify plants by their scent and they get names. Right, and so really, what that name means is a scent profile. So when you hear that name, you expect that the plant's going to have that scent profile. So I'm down to California in 2013. This company's asked me to start figuring out some breeding things for moving CBD around, and we want to be working with scent. And so, in order to really breed for scent, you have to be able to accurately measure scent. And up until that point in time, really what the labs would do is they'd calibrate the GC with like pinene and assume that it was calibrated for all the terpenes. And of course, that's like not a good way to do things. And so our chemist Matt Giese developed this method. And published this paper here, which is development and validation of a reliable and robust method for the analysis of cannabinoids and terpenes in cannabis. And he, this was a lot of work, them doing this to calibrate labs, especially at the beginning when they weren't selling like these very accurately mixed terpene mixes, terpene blends, to calibrate machines. Calibrating for 32 plus terpenoids is a lot of work, because you're plotting every one of them on a curve, and you have to do a calibration curve for every terpene, and it's just like, trust me, it's a lot of work. But in the process, he figured out, okay, now we have an accurate way to measure all the different terpenes in a given cannabis sample. And in doing so, we then had to figure out a way to present this information to the world, right? All this data that we're collecting. It doesn't really do you much good when you get like someone hands you a spreadsheet table and it's got all these words like beta caryophylline and humulene and farnesene, and they're weird words that a lot of people have never seen, and then you've got a number beside the word. It's hard to turn that into like meaningful information in your brain. So what we did is we came up with this color, this way of colorizing a bar graph that allowed you to understand, like once you understood the names of the terpenes or the terpenes responsible for each graph, and we tried to keep it easy, like, yellow is limonene after lemons, blue is beta caryophyllene, the purple is linalool, which is colored after lavender. Obviously, the pinenes are green. Myrcene is kind of this maroon color. And so what it does is it turns that information on a graph or on a spreadsheet into a picture that your mind can understand and decipher once you understand what the colors mean. And that became really useful because we could, just at the glance of an eye, you could see what terpene profile, the plants fit into. So now we've got our lab report format. We've got our way of measuring terps. We're off to the races. We're growing like thousands of plants. We're buying samples from all over California and running them through our lab. And we realized that all of the plants, call it 99% of the plants, fall into these 6 primary categories that are governed by one of these dominant terpenes. I'm going to give both the company I work for, Napro, but also SC Labs here a little shout out. This stuff I stole off their website for the presentation, but it just shows the varieties that are in what we call the pinene dominant class or the limonene dominant class, beta caryophyllene dominant class, the myrcene dominant class, or the ocimene dominant class, or the terpinolene dominant class. And so what we did is through growing and analyzing thousands of plants and thousands of samples, we ended up with a whole bunch of data. Okay. And much like if you had, call it like, say, 3 or 4 decks of cards, say all the decks of cards are, they're regular decks, but the one card has blue bicycles on the back, another set of cards has red bicycles, another set of cards has yellow bicycles, another set of cards has green bicycles. They're all shuffled up together, intermixed. And so what we started to do was just by the terpene color graphs, we started to put them back into their suits. So, we put all the diamonds with blue bicycles together and the hearts with blue bicycles together. And we started kind of just laying all these kind of try to unshuffle these, these suits and put them back to how they came when they were first out of the deck. And in doing so, we did the same thing for terpene profiles. We grouped all the pinene plants together, all the limonene plants together. And what happened was these classes that existed in the species kind of revealed themselves to us. Okay. And we were sitting around at an Emerald Cup one year, and I was talking to the guys from SC Labs about it. And, we were saying how if we were going to, how we thought a cup should be run was that, like in a dog show, the shepherds are judged against the other shepherds, and the golden labs are judged against the other golden labs, and the Dobermans are judged against Dobermans, and the Greyhounds are judged against Greyhounds. And every breed is kind of judged against its cohorts, which is the best one. And it would be cool if cannabis was judged in the same way. And so they eventually licensed our lab report format, the Phytofax, and they started using it for their lab report formats. And they took the classification system, and rather than present it to the public with dominant terpenes, they came up with what they called the dessert class, or the OGs and gas, and the sweets and the sweets and dreams, the Jackson Haze. These things really match the terpene profiles that we were working with before. The tropical and floral plants were the ocimene plants. The Jackson Hazes are the terpinolene plants. The sweets and the dreams are things like, obviously, Blue Dream and your Cherry AK, Perps, Granddaddy Perps, the classic blueberry. Skunk One actually fits into the sweets and dreams class as well. OG and gas are like limonene, beta caryophyllene, and myrcene. The desserts, your cookies, they're like usually high in limonene and beta caryophyllene. And then we've got a few extra special classes that are more rare. I used to call the citrus class the rainbow class. You got your, grapefruit and tangy and mimosa. Some of these things all fit into that class. And then we've got this exotic class, and I think that they've made a mistake here on which cultivars are in it. But you've got like your more floral, exotic-y, spicy plants. And they're usually a combination of myrcene and pinenes and limonene and terpinolene. Again, both of these are more rainbowy classes. Some of you will go back later and you'll actually notice that when you got like the little head and the hair on top of it, when you zoom in on these things, you can see the types of spreads that fit into every class, the types of terpene profiles. So go and have a look at those. Okay, so we want to breed for scent, and we're starting with terpene classes. And so I was going through, essentially all the library of plants that I'd collected off of, the market in California and things from my own collection and these seed lines that we were growing. And you'd find a plant and you'd want to understand how that terpene profile was inherited. So the easiest thing to do was to self it and to cross that plant to yourself. And that way you're minimizing the experiment to the 2 chromosomes that are present in the plant. Okay? And so we self-train wreck, and this is an example of what happened. Train wreck was it's a terpinolene dominant plant. Looks very much like most of the jacks and the hazes. But in the offspring, we grew 100 offspring. 75% of the offspring looked and smelled like train wreck, but 25% of them had a limonene dominant terpene profile. This isn't the exact profile, but it's kind of representative of what came out of that class. But the point is that it wasn't terpinolene. And if you notice the number of plants, the 75% to 25%, we again see Mendel's ratio, right? And so, again, we make F2. I may start making further crosses of these. The 25% limonene plants, when I cross them together, they're true breeding. And a third of these plants, the terpinolene plants, when crossed together, are also true breeding. So now we figured out how to lock in terpene profiles. Well, we figured out 2 things. One, that terpene profile itself is a heritable characteristic. But we also figured out that like all the terpenes kind of come together in like a what I call a cassette. We weren't sure whether they were going to be inherited individually, but it turns out that they more or less come as a set. So we know that terpene profiles are herited as a category, and we're starting to figure out the dominance and recessive relationships between them. And now we can create plants with at least consistent terpene profiles. Okay, so why did the CBD Renee and all these other plants like the CBD skunk and CBD Nordal or whatever they were, why did they not resemble their namesakes? So they were simple monohybrid crosses that focused only on the CBD content, and there was no further breeding to see if they could fix the terpene profile or the scent profile back into the plant. It's kind of like a Sherblotto thing where the breeding project is like you cross the 2 plants together and you see what comes out of the end of that cross. And that's like a cross is a breeding step or a breeding experiment. It's not a breeding project. And what ended up happening is that you got all these undesirable hempy secondary volatiles that come from the hemp CBD germplasm side of things, and they're brought into the cross. And we didn't have any further recombination breeding, F2s or anything to see if they could remove those undesirable volatiles. But what we figured out is that we could do this. We could create a plant with a given terpene profile, or we could take a THC plant with a given terpene profile. We cross it into a CBD plant. I would shuffle that generation with an F2 or maybe an F3 or a couple of selves. And in the process, I could create a CBD version of that same plant. So it looked the same as the THC plant. It smelled the same as the THC plant. And it had the same terpene profile. And because I could create the CBD plants, we could then cross the CBD plant to the THC plant and get that intermediate type, or what we call the type 2, which is the terpene profile plant with both THC and CBD. Okay. And of course, we didn't just do this in the pinene class. These are pinene class plants, but we made them in every flavor class. Like we made type 1, 2, 3 plants in beta caryophyllene class. We made them in myrcene class. We made them in terpinolene class, limonene class, osamine class, etc. And that is actually a really useful tool for not only for formulations and extracts, but it's a really useful tool for doing scientific studies, because there's a whole bunch of these scientific studies where they're trying to show the differences between like THC cannabis and CBD cannabis, but each of the varieties that they're using for that test are they're from plants with different terpene profiles, right? And so in making all these different lines, we essentially created a set of tools for researchers to be able to compare THC plants to CBD plants, or THC plants to THC and CBD plants, um, while maintaining the terpene profile consistent, right?
This is
And
incredible, by the way, Ryan. So are you just saying you just kept going through into the F2 to find that ratio in order to find these terpene cassette decks? Is that kind of the basis?
that's exactly it.
That's unbelievable. It's like complicated, but it's actually quite simple. It's like just keep going. You stop too early. Like people when they were pioneering across Canada who thought Winnipeg was the end.
Yeah.
Ouch.
Well, and it's that's the thing. Like there's, you know, again, I'm not trying to take shots at people in the industry, but it's like the people that are making these crosses, they're not trained plant breeders, so they don't really know what they're doing. They know that they got to cross and make new things, and they think, well, I just cross it together once, I'm creating something new, right? But you know how, and I know you're the same way. We're if somebody shows up at your house and they cram a bag of like not really nice dope under your nose and they say this is Renee, you say to them, no, it's not, right? It's like we
Yeah.
can recognize these plants by their scent. And part of when we get all hyped up about a variety and you're trying to share it with someone that lives in another country or like way across, even way across the continent, the name travels, but what happens is the name gets put on a different plant, right? And so now we're talking about the experience or even the therapeutic value that a plant gives us, but we're really talking about two different things. Um, and when you're making a product, like Marcus, if you're making a special hash product and it comes from a specific cultivar, a plant within a population that has a unique scent, that's the experience of that plant, right? And you don't want to have something else with the same name in a different market, right? It's like that. It's really important to your brand identity is that the genetic is actually represents what you're trying to get out there.
And all you really have to do is plant those seeds from your into your F2, find that ratio of the different ones, and then use the one that has the profile. Like,
Yeah, and sometimes
Exactly.
it takes an extra step. Like sometimes you might have to go back to the THC plant to reinforce the traits, a secondary scent mark profile and shuffle it again. But it's that's what plant breeding is. It's about doing these it's about knowing the steps that you have to take and then taking the steps to get to your defined goal. Right? It's not about just mashing two plants together Sherblotto style and
Yeah.
calling it something new.
Yeah, and it's taking the critical thinking that you're explaining because like if you look at Mendel's work, it was like what ignored for 35 plus years or 30 years or something before it was discovered and or rediscovered, I guess, in the 1900s to be then finally put into practice because it's really it's critical thinking at such a high level that it's hard to understand that you're gonna have to be okay with 86ing and killing things like immediately because they don't fit the actual why behind what you're doing. And most people have a really hard time doing that when they're breeding, just making seeds because they're like you're saying, I'm going to take Sherblotto style. I'm just going to take this F1 and this air quotes proven plant, and I'm going to make this new thing. And that's not to your point that there's more steps to this to actually create your why and to actually get after that reasoning behind it to end at a result that actually is accomplishing the goal.
Well, and don't
And
get
that's
me
just,
wrong, like
you know,
mashing plants together leads to some incredible discoveries. Like
sure, for
that's
sure.
all how we found all these new scent combinations. It's just that once you have the new scent combination thing,
Yeah.
when you're naming a seed line after that plant, what you should be doing is trying to create the seed line so that that plant shows up consistently in the seed line.
Yeah, no,
And
and don't get me wrong. I'm not saying it's not good to make outcrosses and, you know, do that. But I do think, to your point, what you've been illustrating all along is that if you do walk this path and do the work, you're going to come out with results that are superior in a lot of ways.
yeah, and it's just like, again, I'm trying to give people the set of tools and understanding so that now they can apply that to their breeding programs and say, hey, I've got this thing, rather than just crossing it to cookies or OG or sherbet or whatever the hype thing is of the day.
Yeah.
It's okay to take a couple of generations to try to figure out, hey, how are the traits that make up this thing that I like, how are they inherited, and can I put them together in a seed line in such a way that the seed line matches this special plant,
Yeah.
right? And that's really the thing that's missing in today's seed industry, unfortunately.
Do you want to continue, Ryan, and then we'll get into more of the discussion just so you can finish off?
Sure, sure.
Sounds good.
So yeah, so this is, now we've got terps figured out, we've got cannabinoids figured out, and we can essentially custom mix and match them in any given plant. Okay, and we've made all the terpene types and all the flavor types together. So we've really got a lot of stuff to work with now. Okay, okay, but we know through all this recent work that we've been doing, and Mark having lovely folks like intelligent folks like Mark Sheldon and the abstract guys coming on the plant or on the show and talking, that there's all these other flavor molecules that are also going on top in cannabis on top of the terps. So I say cannabis scent is an orchestra of volatiles. And really what we perceive as scent as one trait is comprised of layers of scent and flavoring molecules, including the terpenes, thiols, esters, and aldehydes, right? And so within the terpenes, we've got the monoterpenes and the sesquiterpenes, and then the modified terpenoids like linalool. We've got thiols, the sulfur-containing terpenoids, prenylthiol or 1-MBT, which is the skunky terp or skunky note. Esters are this new class we're learning about in cannabis. They're like also found in all these different fruits, like these exotic fruit lines. Same molecules found in cannabis, and then this aldehyde layer, which are also like they're found in cannabis, but they're also found in like, when they're making perfumes, they add these things to perfume to help them bloom, right? Help the scent kind of evaporate, or mature in the bottle. And the plant also produces these things, okay? And so here's a list of some of the esters that we found in cannabis. Not the cannabinoid esters, but specifically the fruit ester, the same molecules that are found in the fruits. We've got ethyl hexanoate, which is like fruity. It's fruity and sweet. It's found in pineapple, banana, green apple, strawberry notes. Hexyl butyrate is really like, smelled to me like Vicks Vapor Rub, but some people also say that has like apple or pear notes. Propyl hexanoate is found in pineapple and blackberry. Ethyl caproate found in other exotic tropical fruits. And then this methyl anthranilate, which is an interesting one, is found in Concord grape and bergamot, but it's described as like grape soda. Oops, spelling mistake there. Grape soda or candy. Okay. And so now, remember we were talking before about when we're breeding, we want to be able to measure these things because we want to look at like plants that maybe have varying doses of these genes. So now these are new molecules that we can start analyzing for in our GCs, right? And seeing if we can measure. Because if we don't measure it, we can't manage it, as we know. We've got the cannabis aldehydes. Decanol offers a powerful kind of sweet orange peel aroma, also used to enhance like perfumes and soaps and shampoos. Nonanol is a natural fragrant used in trace amounts to add bloom or enhance floral and citrus notes in scented products. And then undecanol has a fresh citrus scent used as a scent enhancer for other consumer products. Right. So these things that we're finding in cannabis, We know they're used in other scent products, and we can try to optimize or enhance these in the plants through breeding to kind of improve scent, right? And of course, we've got the whole thiols or the cana-sulfur compounds, or what the abstract guys are calling the Tropicana sulfur compounds, which I think is a bit of a mouthful of a word, but these words are also mouthfuls, as you can see. Mercaptohexylacetate and mercaptohexane 1-all. Sheldon will be here telling us all the things about these because he geeks right out on the chemistry. This one prenyl thiol or 321-MBT is like the key odorant for the gassy or skunky notes. It's also what we find in light struck beer. So the point is, we should be able to start breeding for these things too, really to be able to target scent. And in fact, Marcus sent me a link the other day on LinkedIn, and he was this guy from a group in Eastern Canada. They've actually shown that there's some linkage markers or genetic markers associated with some of these scents. So even through those markers, we'll be able to use those for driving breeding programs and, really complex creation of scent profiles through directed manipulation of these genes and pathways. Okay, so again, just to kind of hammer home, it's the cultivars that are really the thing behind your product you're putting out on the market. The cultivar is kind of like the genetic recipe, but how that final dish turns out in the package is really what the cultivator optimizes through managing the environment. But the cultivars are critical, and I don't think people really give that kind of attention to the industry yet. They should, but we're not quite there. So here's something else we can do with breeding. We all know Wade did this great work with children and that had epilepsy and all these different needs for specific conditions. My approach down in California was, can we make a better oil for the children with epilepsy? And, I'd been looking into, I go to these scientific conferences to talk about cannabinoids and what the science is showing, and we realized that CBDV and CBG have both shown promise in intractable childhood epilepsy. And so the idea became, can I create plants using these CBDV and CBG plants that are better or maybe augment the CBD oils that they're using for children? And so I did a big deep dive literature search into all the terpenes, looking at their pharmacology, realized that beta-caryophyllene specifically and linalool each have antispasmodic and anticonvulsant properties. Linalool binds to the same receptor set that the benzodiazepines like Ativan and Valium, they bind to the same receptor molecules. So it kind of has this anti-anxiety effect on the body. And it's been shown that it also has antispasmodic and anticonvulsant properties. So, using our kind of this entourage effect theory, the idea was, can we create a CBDV plant or a CBG plant or both and breed in linalool and beta-caryophyllene? See, I've made another mistake here on the bottom. Spelling mistake. But yeah, so we undertake this project through hybridization. I've got a THCV plant that's high in myrcene, had a terrible volatile smell. It was truly hemp, super tall, thin plant, terrible flowers. It was the ugliest thing you've ever seen. But it had THCV. And so we crossed that plant into a CBD plant that I had engineered to have linalool and beta-caryophyllene. And the first generation, of course, everything smells like the crappy myrcene plant. None of them had the beta-caryophyllene scent profile that we wanted. And all of the plants in the F1 generation had not only THC, they also had THCV, CBD, and CBDV. So at least we've now got our plant that has CBDV, but it's got too much other stuff in it. So we, again, wanted to self and create an F2 segregating generation to see if we can recover plants that have no THCV and also recover plants that have the terpene profile that we want. So we make our F2 crosses. We selected the best of those F1 crosses or F1 plants, created a couple of populations of S1 generation seeds, which have the F2 segregation pattern, and we screen those plants using TLC again, and we're going to destroy all the plants with THC. And so this is what happened when we cross those when we cross the plants with all those different cannabinoids together or self them, we ended up with these these 6 types. Only 12.5% only had the CBDV and the CBD. And so all the plants that had THC in them, which is these top 4 rows, we got rid of those by using thin layer chromatography. So that was going from
Were you roguing?
we were roguing. Yes, at this point in time, we're
I'm
roguing.
learning.
Yeah. So this was not a small week. Like, I'm going through the greenhouse, cutting down like leaves off of every plant, taking them back to my little lab in the head house, adding a little bit of ethanol to each leaf, kind of crushing them all up with my little crusher, wiping off the crusher with alcohol so I can clean it before I go in and mash all the plants. And I essentially make 300 little extracts. And then we put them on TLC plants, and I get this data back and realize that 12.5 of the plants have the CBD, CBDV chemical signature that we're looking for. I kept notes of everybody else, eventually went through and rogued them all out of the plant, killed them all. And so now we're left with our 12.5% of the population, which is not much, that have CBD and CBDV. We carry those plants through to harvest, and I could tell by nose, but we sent them off to the lab and we got back the data. And again, 25% of them were beta-caryophyllene, 75% of them were myrcene. Again, that's our 3-to-1 Mendelian ratio. But when you multiply 0.25 times this 12.5% number, we end up that only 3.125% of all the plants that we started with had the dual set of chemicals that we needed. Okay, so we're going to take those plants and work with them again. So we cross those things back to the CBD line to see if I can reinforce the terpene profile. And we also self them again to create an S2 generation to see if we can, help reinforce the CBD, or maybe we can get rid of the CBD entirely from the hemp or from the CBDV line. And we've got a couple of populations that we can cross together to kind of restore some of the vigor from the inbreeding depression for the traits not under selection.
Yeah.
So we end up with this very special plant here on the right, CBDV. You can see it's high in beta-caryophyllene, and linalool is the third most dominant terp. And so eventually, it's not on this picture, but eventually I made one that was CBG as well. And so now we had CBG, CBD, and CBDV oils that all had the same terpene profile and scent profile. And so those things could be passed along to the doctors, and they could use those on clinical trials, right, for running to see if we made an oil that's better for kids with epilepsy. So that was a really satisfying project. I mean, it had no recreational fun. For most people in church, it's like, who cares? There's no THC in it. It's not a very exciting plant. But it was actually like a really cool thing, because this was like really the first time that anybody had taken a plant or had created a plant out of an idea. Normally people were like, hey, what condition can we match with which cultivar? And like the plant breeder approach is, can we optimize the chemicals in a cultivar to really match a certain genetic condition or a therapeutic need, right? And this was really the first time that had been done. So I'm quite proud of that one. And then this is just some ongoing work, same kind of thing that we're doing. This Victoria Cannabis Company, a friend of ours for a long time. They're in the cannabis market. Everybody wants the high THC plants, and they've got this G-Wagon plant that is like 34% THC. And so we're going through a series of breeding programs, crossing that plant into other plants to see if we get other plants with known scents to essentially harvest that one chromosome out of the G-Wagon that's responsible for the super high THC production. And can we move this thing into these other scent classes? Really, this is a market-driven kind of project. It's the distributors demand such high THC content all the time that it's just a need. So, we're doing this to see if we can make some real high power stuff. And of course, this is a cross of the G wagon
No,
to the After Eight, and it's already in the F1
no
is already
problem, Cal. I'll go get them.
the first generation is already 27% THC, but through selves we should be able to pull out some plants that are in that extreme high THC combination, and if not, we'll back cross it to the G wagon and resort again. So yeah, that's the kind of thing that you can do when you understand how to move traits in and out of populations, and you're just making crosses and going about like a scientific method, and in the process you should really be able to like recover the things that you're looking for. Challenges that we run into inevitably is cost and budget. Lab testing is expensive, so we try to leverage technologies that help us minimize the cost, like the thin layer chromatography, or we can now use these infrared, near infrared, or FTIR infrared devices. Understanding the Mendelian plant ratios and keeping meticulous notes and being able to recognize the patterns is the most difficult part about plant breeding. But it's definitely not past any one of the people watching the show or on the panel. It's just taking the time, making the notes, recording the number of individuals with each version of a trait, so that when we have all that data at the end, we can kind of look through it and figure out which are the plants that have the right genetics that we're looking for to move our projects along forward. And I think that's it. There's some like how to reach me for seeds or questions, or I'm Chimera Genetics on Twitter. That's more me ranting and raving about stuff unrelated to cannabis. The breeding cannabis Instagram I try to keep for cannabis specific, for helping teach some of these concepts. So you can
Dude, this is such incredible stuff. You shouldn't even waste another minute like moaning and bitching on Twitter to people. You should just be posting shit like this, because this is really good stuff, Ryan. This is, and it's, I've been involved in cannabis for a very long time. I didn't know a lot of this stuff. I understood that doing it right is how you had to go about doing it. I'm glad you brought it back to that one, because that was the one that blew me away the most. That whole like, look, you can't just cross the sherbet and the gelato and get the sherblato. You're going to get a whole variation of 25%, 50%, and 25%. You actually got to get into the F2s to help, well, the idea of moving the terpene profile from a THC or a THC CBD cultivar to a CBD only cultivar. You really do. I truly understand the medicinal value of that beyond the recreational value. It's super important work.
Well, we didn't know how that was going to turn out. Like, we didn't know if the terpenes would all in like that cassette, that profile was a heritable trait, or if like were the terpene profiles going to blend together, right? And so it was a little bit of a surprise. I mean, it wasn't totally a surprise. It was one of the options, but it was just neat to see that they came out as a cassette, because it really meant that we could hijack and kind of cut out that terpene profile from one plant and move it into another plant. Obviously, with the other layers of scent, like the thiols and the esters and the aldehydes, it's a little bit more complicated than this. But it is just another layer, and it will end up inevitably being that we can also breed those layers on top of this one, right?
Well, yeah, there's a rabbit hole that you're going down, but Mendel's law invites that critical thinking through data, right? And I think that's what's in good to highlight here, is that you utilized and leaned into the data, but then had that leap of faith and an exploratory moment, right? Let's be real, because there's a moment of discovery there, right? As you're highlighting. But it's important to not discount or minimize the data that you collected.
We couldn't have done it without having the ability to collect the data and measure the terpenes, and the
Yeah,
lab report format, and the ability to turn these terpene profile numbers into a picture that we can
right.
identify. Like the
Yep.
human eye is really good, or the human brain is really good at like identifying things through pictures, but through like when it's just lists of numbers and words, it's like sometimes the pattern gets lost in the noise. And
True.
so this
Yeah.
was like a really helpful step, the phytofacts. And I think it's a really uniform, a really nice way to be able to give that information to the consumer. You still have to engage with them and help them understand what it is, but once they know how to read the graph, um, it's a really powerful tool. The funny thing is, I don't know if people have noticed, we have the same, like the top three dominant terps, like the light green, the darker green, and the purple. They're actually the lines that are on the top of the graph of the phytofact always. And so what we're kind of doing is like you're subtly implanting that information into the brains of the people who aren't even looking at the graph, right? So eventually they get to know, oh, I like the ones that are green and purple, or I like the ones that are yellow and blue.
It seems like there's an infinite deep rabbit hole to go down here if we can create and invent new machines to perceive things that maybe we're not perceiving, and maybe younger people to come into the space who think outside of the box, who can see patterns that maybe the past folks haven't seen. There's so much going on here.
Yeah,
You
it's
know, the,
multi-layered.
the
Like the machine learning aspect of it comes to mind is like that's where I think because this in my just listening and knowing, how much work goes into this, it's really visualizing patterns for us then to like as you're saying, Marcus and Ryan, to take the other layers of it to further help people understand it, right? And that it's just the very first step of the process and I think history repeats itself. I think a lot of things are being misunderstood right now in terms of data and how to utilize it, much like Mendel's law, what took 30 years to be adapted, right? So I think there's a real opportunity here to take all of this that's been what this has been developed what almost how long ago, Ryan, did this start?
well, this is 10 years ago now, 11 years ago.
Right. So we're, I think there's a lot to learn from history here, and if we could just perhaps take a lot of these concepts and continue utilizing them, maybe there's some more paths to unlock.
Oh, 100%. But there so are so many paths, and that's really what I'm hoping with, kind of reinforcing the importance of some of these things is that some of the breeders will go back and be like, okay, I need to go back and learn the Mendel stuff. It's not that hard. It's really like
No.
this stuff is not that difficult.
Yep.
It's just taking it and taking the idea and the understanding. Because like, look, Mendel did this work before we even knew what chromosomes or DNA was, right? And then DNA came along, and we understand chromosomes, and now we know that the heritable factors are on the chromosomes, right? And so the alleles are just versions of a gene, and it's like we've built on this whole understanding. Right now, talking about training AI models, we've got genomic sequencing. We can tie all of these phenotypes to a unique genetic thing that we see in a genetic sequencing event. Okay, so because genetic sequencing is becoming so cheap, we're going to go through a phase where we're figuring out the associations between these traits and the genotypes. But soon we'll be able to track just the genotypes and not even have to look for the presence of a thiol. We'll just know that that thiol will be in the plant because it's associated with a particular genetic marker, right? And AI and tying AI and machine learning, and I have friends that are doing that with genomics. That's the next thing. That's the next level thing. And so we're really going to start getting to a point in cannabis where you can make the crosses, you can plant 1,000 seeds, you sequence all the plants. By the time the plants are like their third or fourth node, we'll be able to come back and throw away like 95% of the plants because we'll know that they won't have the right combinations of traits that we want. And then
Right.
you grow out that last set of percentage plants to get the full evaluation of them. But it's where we become really efficient with our time and space, and we can really start making some progress. And we're kind of on the cusp of like all these really interesting things. Like, like I said, they found the first marker associated with that canosulfur thing. It's going to be coming out in a paper soon. So, we're building we're building the plane as we're flying it, but the plane is turning out pretty damn cool at this point in time. So
How does
it's
Linnaeus factor in with like Mendel?
well,
And
Mendel was, I don't know if you saw that early slide, but Mendel, we've got the name of the P. Where are we back here? Right here, right? Pisum sativum. So that's the Linnaean. Anytime you see that L, that means that's the Linnaean name.
okay,
And you probably recognize sativum is pretty close to sativa.
yeah,
Sativa is the feminine form of that word, but can't sativum just means cultivated or useful, but it was also used as cultivated. So it's like that's the pea plant that they cultivate for the useful purpose of feeding the monks. So a lot of that, this all this stuff was going on at the same time. Like I said, like this was 1856 through 63. On the Origin of Species was published in 59, right? So there's a lot of people around the world that are kind of having these same thoughts about like how do species evolve? What is a species? Are these traits heritable? Can we move them? It wasn't until
well,
1909 that the Germans figured out how to harvest nitrogen from the air, right, through that chemical reaction. And that was really what allowed agriculture to take off, right? Because it's like all of a sudden now we can produce mass amounts of nitrogen, right? And so it's all happening at the same time as we're learning how to breed plants only really 100 years ago. And all of these things happening at the same time is what led to the Green Revolution, where now we can grow crops by the thousands of acres and feed the world.
yeah, but not that healthy, right? I mean,
Well, it's different. It's not as ideal as the way that you can do it on a small scale. The
it's chemical growing.
the argument though is that we can't feed the world doing it all the way that you and Kelly do it, right? I mean, if everybody dedicated their lives to growing their own farms, sure they could feed themselves like you guys do. But that's just not realistic for a lot of people, right? Because they're living their daily grind. So we have
I
to
would agree
turn.
with you. The modern era has brought us away from that. But when you look at like the victory gardens and all of the stuff that's been proven over the course of periods of history, like we can absolutely produce our own food. It's just not in this modern system. And what you're saying is correct. Like people are not, they're not, there's not an agriculture anymore. It's a modern culture. And so we have to depend differently on agriculture.
Yeah, we're way out of scale for doing it that way. You can
I
do it that
wonder.
way on like an artisan level, but it's can't feed the world that way.
Well, I wonder, is
I
there
wonder because we waste a lot of food. We waste tremendous amounts of food. But go ahead, Colin.
well, I was gonna ask the question to the group, like, is there a way to take well thought out selection, i.e. Mendel's law, through a selective process and utilize that in a system, call it, these systems, because let's face it, the way that we cultivate our systems, right? We'll call it organics, call it what conventional, whatever you want to call it. These are, at the end of the day, they're all just systems that are getting to an endpoint. And is there a way to instill some of these practices to better these systems so both sides can benefit, and perhaps you don't need to use conventional systems, i.e. Large scale, Ryan? I don't know, I'm just asking the question. Could that be a thing that we strive for? Because I know that we all are after sort of the same thing. It's just the perception of how we get there and what system we use.
But I never saw anything about how many marijuanas it's going to take to get me high.
Depends on the marijuana.
It's in the allele.
It is. It is all in the alleles that the plant has. Funnily enough. Right. And listen, we also figured out stuff I didn't put in the presentation, but I mean, I talked about it on a podcast a few weeks ago. They asked me what the weirdest thing that we ever discovered with breeding, and in our CBD breeding, we realized that we could make 2-to-1 THC plants, and then in trying to inbreed those, we accidentally discovered 6-to-1 THC to CBD plants and 8-to-1 CBD to THC plants. And that particular ratio had never been seen before. Like it was either, like 15 to 20 to 1, but there wasn't really one where it was a lot of THC with just a little bit of CBD. And that little bit of CBD it takes the kind of the edginess off of some of the high. So for certain people like those, I think those plants would be useful. It's just the market doesn't seem to. It's more the distributors don't understand the importance of those things, and I think
Not yet.
not yet. And
Not yet. But Marcus, I mean, I've heard Marcus and I've had the conversation about like one day I think people will start to realize they just want something that tastes extremely good that they could have all day,
yeah,
right? Rather than, you a couple hours and then you're just like, I need a break all day. Like how do you keep, a repertoire of things around that taste extremely good but don't, couch lock you, because that's another thing that I find a lot of consumers complain about, but then they go back to the store asking in this vicious cycle for high tack,
that's the thing.
and it's
That's the
like
thing.
I
It,
get this question all the time from people like, hey, why aren't you guys making products that are like less tack? And I'm like, well,
nobody buys them.
dude, then go into your shop and ask for that,
No, that's the dichotomy, right? It's like even
because
the
it's
people that think they want it or maybe understand that they want it, it's like they get into the shop and they're just drawn to that high THC. What's the highest THC, right? And it is, as
it's
we know
confusing.
from like the whiskey and the wine world, that's like the strongest one isn't the best one. But it's not only and in Canada, like the distributors have seen that it's the high TH stuff that sells, and so now we've got this like positive feedback system for THC content to the point where I'm like I showed you in the last breeding. The ongoing work is like, how do I take these plants that are 25% and make them 30 because 25% isn't enough, right? For them to be like popular in the market. So it's like the market demands the high THC ones. So we're kind of going back and feeding the market what it wants.
Well, that one plant that we always talk about that had the
The
you,
Valencian dominant,
the
yeah.
Valentine dominant, that came from Alan Capular and the Peace Seedlings family in Corvallis. If anyone doesn't know who Alan Capular is, I mean, he's an amazing, one of the groundbreaking genetic molecular biologists in food, mostly. And he got one of the highest grades that Yale ever has received at the age of 16, and he was working with chromosomes of orchids, and he combined colors and did different things. And he's a spectacular human being. Now passed away. He was known as Mushroom. He was a cult classic from Oregon, from the East Coast, and the Peace Seedlings children, Delana, we did a lot of stuff at Country Fair together. We did the whole Living Willow fence at Oregon Country Fair together. They have an amazing, and that's where kinship gardening came from. And I love the idea of planting gardens, based off the families that they come from, and knowing the whole family tree and from the ancestral seed to where we are today. And
Well,
Alan,
Josh, we got to go back to them and see if we can recover that plant
I
because
know.
that's such a like I didn't put it in this breeding presentation, but that's in a level that plant is in a level of rarity that is just it's so unseen or unheard of. It's like the Selenodiene plant. It's like the white thorn rose.
Okay.
It's like it's a white whale,
What was
right?
weird about it is it had the variegated ruffle leaf on it. So, and it came not from the freak show. And I had numerous arguments with the freak show guy, or online about it. And he was like, I don't know, blah, blah, blah. And I just got like, I'm just like, This came from the pea seed family. It's a rarity, and it just grew, you know, right? And we tried crossing it, and it went away, just like you said. The whole trait and everything went away. No more ruffle leaf, but the flavor did come through on some of the crosses. But so not only was it a rare plant, but it had the whole crazy ruffle variegated leaf. I have all that documentation. So anyway, yeah, the peace seed family is continuing to breed cannabis as well, but they don't have machines and they're not working with Oregon State University anymore.
No, but
They're just
maybe those things are hidden in the genome, and that's what happens with things like that leaf phenotype. They're hidden. These traits are buried as recessive conditions in other genome populations. So it's like you don't see them until you do. They just pop up, but they're in there. So it's funny when people get all upset about, hey, that's my trait, right? But because again, it's like, as we just showed, it's like you can move those traits or you can separate them from the scent, right? So that's Mendel's second law of independent assortment, right? So it's the understanding that we can take these traits from varieties and kind of disassemble them and use their building blocks as like ingredients to put back into other seed lines, right? That's what breeding really is. So
one thing that was unique about Alan Capular is, and you can look him up in any form that you want.
you showed me I've still got a video up on my computer of him.
Yeah, there's the video, and then I have the whole handwritten file on amino acids. He did a whole entire GLC and HPLC thing on amino acids and foods. And he was trying to raise the amino acids and make food more nutritious. And he had like 200 different tomato species at the time on his farm in Corvallis. And he was studying all of that because he knows plant breeders across the world. And but he chose open pollination and public domain as his like way to move forward, and he didn't really agree with hybrids and monocultures, and he thought he was that there was a fragility in the sameness of everything, it resonated with us. And again, it's not one's better than the other, but he just chose this diversity platform, with his plant breeding, and I just found that he's just such a fascinating person. So it was so interesting to speak with him on that.
Yeah, well, diversity comes at the expense of inbreeding, right? And uniformity. And sometimes if you're trying to lock down a trait, the only way to do you can't go with diversity, right? And so it's like it's really balancing those two forces and going back and forth between the two, right? You want to inbreed enough that you're capturing the traits that you deem important, but you want to outcross enough to be able to maintain the genetic diversity not at the loci under selection, right? So that's part of inbreeding. Why you do when you are inbreeding, I don't know if you saw the presentation, but in that third and fourth step of the CBDV line, I was crossing the individual inbred lines back together, right? And what that does is it allows me to fix the traits that I'm chasing, but restore the genetic variability at the traits not under selection, right? Because it's like I was saying a few episodes ago with the crack house genes. It's like when you're selecting for something so tightly, you end up with the broken or the defective versions of genes that are on chromosomes or are nearby on chromosomes, right? And so that's the balance, is like inbreeding to fix the traits that you want, but outbreeding enough within your family so that you maintain variability at those traits not under selection, so that you can still maintain and fix all those traits in the population, right? So it's one of the always like ever existing challenges in plant breeding. How do you do both, right?
Welcome, Caleb. How are you doing, brother?
Doing well, doing well. Ryan, is one of the answers to that question, how do you do both in separate populations?
You're muted, Ryan. I was muted trying to tell you you were muted, by the way.
Yeah, it's separate populations, right? And then you try to bring them back together. Um, it is the balance, right? Because it's like sometimes, like for example, like the Selenidiene or the Valencian dominants, there's no way to get those things and make them uniform without inbreeding. So how do you do that and maintain genetic diversity? Right? Like you have to be paying attention to both things, because like Josh said, I think there is a detriment to only doing the inbreeding, right? But if you're not doing the inbreeding, you're also not finding the selenidiene or the Valencian or maybe like those high amino acid nutrient plants, right? Like it's kind of the breeder's dilemma, and something you got to be paying attention to as you go about your breeding projects. So.
Welcome, Dr. Mark. I see you're not on camera, but I'm still going to welcome you. I'm sure you'll be around in a bit. Josh as well. Welcome, brother. We've had a long lesson today, 2 hours of breeding school. I hope you all took your notes. There will be a test in the third hour of Hash Church. And oh, you're muted again, Ryan. It's dabbing
After
hard over there.
dabs, got to do our dabs, and then there's the exam.
Yes, exactly. There he is.
Is it a multiple choice?
What I missed? I missed something?
No.
No, I didn't miss anything.
Yeah, I'm gonna give a shout out here real quick. Right. Caleb and I made our way up to Hopland, California for the Culture Cup. So shout out to the Culture Cup folk for having us pop by. Really cool. Local competition. Just yeah, a lot of good shit. Great scene, man. I mean, the people up there all look out for each other, and yeah, it's nice glass there too.
Didn't the rosin winner come from Maine? The winner of the rosin arts and craft came from Maine, I think. If I'm correct. Anyway,
Caleb?
shout out to Maine. Big up. That's where Hidden Forest comes from as well. So it's great. It's amazing. And let's take a minute to recognize like there's amazing people in Maine, like Best Friend Farms, Brian coming from there, Helios, they've done a ton of different things over the years. Professor Q. Wow, there's a lot of people out there, and yeah, Maine's got really good laws, and it's far north, and they have a lot of diversity in the types of things they can put on their farm. And I know that Hidden Forest in particular, Hunter and family, high respects for them, of course. They first got known for at least coming really high at the Ego Clash or something, and it was always sun-grown, light depth stuff, or maybe even full-term sun-grown out of Maine. And then they came out to California and got super high. So they've been making waves, I would say, and I believe, and they're going out to Spain as well. So, and that, yeah, so it's cool. It's cool to see Michigan and Maine and those people like really rising up.
Hell yeah. I got to give these guys a shout out too from I got to put a picture up like the way you did there, brother. Shout out to Steve Aboud and Ruben Valenveras and Alex Goldberg, who I shared my booth with, the Sambo Creek Filtration and the Valenveras booth this week at the Grow Up Conference. We had an absolute blast. And I always look forward to the gatherings, as I'm sure you do, Etienne. Just fun.
I love it. Somebody recognized me from Hash Church. Was like, "Hey, you're DTA from Hash Church." I'm like, "Yeah, hey man, that's good."
You're like, "That's true."
And yeah, I did. Yeah, I had a little Darby piece. I had a Radcraft piece, and I had my hat. So I was running a little dab bar while I was kind of sitting there because I just can't help but share, hash with people when they come by, and I love talking hash and smoking hash with people. So, looking at weed and all that just makes me happier than a shit covered fly. I don't know. It's just something about it because we're with our people, right? It's one thing to get a couple of us in a group, but when you see, quite a few hundred people all getting together, all with their glass, all with their rosins, all with their stuff from all around and just sharing it, smiles and laughter and giggles. And yeah, there's something just really fucking infectious about that. So, I love competitions. I love group gatherings and cannabis. It's just, I don't know. It's just something I've appreciated from back in Can, from, my early days when we were at a wedding and Ben Mazel got up and kind of tapped his thing because we were all in DC and was like, I know everybody here is from all around the country, some from around the world, but I know everybody's got weed in their pocket right now. So everybody's going to take their weed out right now. And we're all going to show it, and we're going to just have a competition right here on the spot. And that was 1993, right? So for me, yeah, I mean, my elders always implored to, and which was funny, what won that competition, I still remember to this day. It was called the Kevorkian because it was Killer Bud. And it was just this amazing cannabis that everybody was, when it went around, everybody got to smell it, and then they rolled a joint of it and sent it around. And that's how, everybody had a taste back then. Now there's competitions, and, like this competition, I think it's those who are in the, and, actually submitted, or the actual judges, like the secret cup and stuff like that. So, it's inherently part of our culture to want to not only taste but to judge, right? To what really hits for you. And it's one thing that's what hits for you. It's another thing when you're in a room with 40 to 100 other people and they all agree that this is the shit. There's something amazing about that, right? That crosses all socioeconomic barriers, tastes, flavors, everything just all seems to combine in a way that is undescribable at a certain resolve.
Well, it's almost an incredible thing because you have a whole room full of people who completely and totally suffer from cognitive dissonance in the sense that their weed is the best. So to have that drop down and the veil lift and everyone agree that this is the word of God, this is the one, is awesome. And there's always going to be a guy in the corner going, yeah, this fuck, it was pay to play. You had to pay them to win, and it's fucking bullshit, man. But whatever, I'm gonna go enter the Eagle Clash, and I should be good.
Yeah, of course. Why not?
It's crazy.
100%.
I never I never got into entering competitions, but I've judged many of them, and I know that people look forward to them. It definitely seems like a very important part of a seed maker or a hash maker's scene, like their business. Like it's you want to list off your winnings, where you represented and where everyone agreed that your shit was the best.
We entered Girl Scout cookies in 2013 and left. We didn't expect to place. We got a call on Monday saying, "Hey, you want to come get your trophy?" We're like, "Wait, what?" It's just random sometimes, right? You don't know what people's universal flavors through a competition are going to reflect.
No,
it's random.
It is. It absolutely is.
Did you see the Mary Jane guys, Marcus, at Grow Up by any chance?
Like Mary Jane from Berlin?
Yeah, the Mary Jane. Okay.
No.
Yeah, they're in Canada right now. I'm not sure if they ended up making it down there, but anyway.
A lot of people, though. I definitely hung out with the what was it called? Canatrol?
Oh, nice. Yeah, Jane and they're amazing.
Yeah, wonderful people, wonderful people. Gave them some hash, had a really good discussion with them, and I'm actually going to have a call with them this week. I really liked his talk. He was talking in a way that like a lot of people didn't really understand. he was bringing up, the way cheese off gases and different meats and stuff, and how he kind of came from that, and a really, really cool, deep conversation on bound and unbound water
Yes.
in cannabis and the difference between the two and which one is feeding yeasts and molds and bacterias and such things like that. So he's going to come on eventually here in the next little bit and share some of that with us. And yeah, just super cool people. I ate
Yes, they
mushrooms
gave
with
us
them.
a custom.
They were like,
They gave us
here, giving us mushroom chocolates, and really nice folks.
They're sweet. They speak about the difference between a dehumidifier and the
Yeah, he
technology.
goes he goes through the whole thing in his presentation. It's amazing. I want him to do the same thing that Ryan did today
Yeah,
on Hash Church, like a 1 hour presentation of just everyone listening and him going through the slides and talking about it.
very
Man,
valuable. And it's also it's adjustable. The machine's adjustable, so if it doesn't come out exactly how you like it, you can adjust the
I've
cycle.
got one on the way.
We have one here, and
I'm
we
gonna
used it
play.
last fall, and we have a custom,
How much could you
drive.
fit in it?
It's of weed, it's like around 2 pounds or something, in the 2.5 pounds or something like that, I think is it.
Nice. Like a kilo.
I'm frozen. I don't know if I'm still talking. So
You're talking. You
there you go.
froze for half a second. Like a kilo, a little over a kilo.
It's somewhere in there. It depends on how fat
He
much.
does he does design rooms.
He does. Yeah, no, that's the whole thing. They have pods, they have containers, they can design a custom room as well, as you said. So
Awesome.
I would totally suggest that for the GMP or for the GMP processor or the GACP farm, whatever your international credentials are. They're really sweet, and they sponsor a lot of events as well in the world. Them and Grove Bags, Lance and Jack are really very sweet on that. Yeah, so we're gonna have a
I
big
actually
space.
I actually gave him some of do you know the guys from Calyx,
Oh
Alex?
yeah, in
So
Oregon.
so he's doing his own bags. He's been working on them for 18 months. They're
Oh,
9 layers, these bags. I'll send you some. I gave some to Jane and David at the conference. I'll send you some too, Colin.
You're talking
Absolutely.
about the cure bags?
Yeah.
Yeah, they're incredible. A friend
Yeah.
of mine was just talking about them the other day.
I'll send you some.
They make the Grove bags like look
Well,
like they have some work to do.
he spent 18 months on this shit before releasing it. And he's the same dude that decided he was gonna make a square glass jar when it was like, no, you can't do that. That's gonna be really difficult. He's just like, I'm doing it.
yeah, those
So
guys are super innovative. I think they washed all of Brandon's Skittles from the farm he did in Oregon
okay.
last year or something, but I think that went a little sour,
He,
possibly.
he
But
helped me in Barcelona at my last Legends of Hash, Alex, in a way that when I really needed someone to help me, that guy stepped up in a huge way. And so I always am thankful for him, and kind of will always support the things he's working on for sure. I know, well, Ryan's not at his computer now, but I know there was quite a few thiol and volatile organic compound pages that probably Dr. Mark wanted to comment on. I could feel your ears burning, Dr. Mark.
Uh-oh. Chemistry?
Yeah, he
Can you
went
guys
over
hear
some
me?
decent chemistry. We can hear you. You're a little quiet, but we can hear you.
Oh, okay. I'm still trying to figure out this stupid microphone. Are you getting an echo when I speak? No?
Only if someone has their mute mic. Yeah, Josh. There we go.
Uh,
You're fine.
okay. Yeah, I was cool. I mean, I was actually I was making gummies and listening to Ryan's thing. Yeah, it was really super informational. And yeah, when he started going through the secondary metabolite profiling, I know about that Giza paper and method. It's super useful. And yeah, great that they actually did all that work because now there's basis out there to basically do that kind of profiling. Um, and yeah, I guess the other interesting thing that you're mentioning TLC is really super useful in chemistry. I use TLC every time I'm in the laboratory because just it's so useful. And it's if you're doing a chemical reaction where you're changing the polarity of the compound, you could use the TLC plate method to basically determine whether or not your reaction's gone to completion. So we use it all the time. The one thing I was going to ask Ryan, and I guess he's not there right now. Oh, there he is. Yeah. So again, the plant makes cannabinoid acids, right? Not these cannabinoids. So is there decarboxylation that's happening before you're doing that profiling? I mean, obviously,
So
again, if you're shooting,
that wasn't actually my photo. Those were just photos I bragged.
yeah,
We're using C18 plates.
yeah. So C18. So we could talk about this a little bit. So C18 is what we call reverse phase chromatography, right? So normally the chromatography where you just use silica gel. Silica gel is a polar material, right? It's basically just ground up sand or glass. You know, it's all silicate material, and so it's a very polar material. So it binds other polar materials and lets non-polar materials go through more rapidly. So at the top of your TLC plate, you're going to see less polar material. The more polar the material, the lower on the TLC plate. So you're using reverse phase. So here it's just the opposite, right?
Yeah.
So the more polar it is, the more it's going to move up the plate. The less polar it is, the less it's going to move up the plate. And so what reverse phase is they've taken the silica gel particles, and so these are C18 plates.
No, that picture was just something I took off the internet.
Oh, that's something off the internet. Okay, so if they're using what's called, sometimes they'll call the stationary phase in the column or the plate C18. And that's because what they've done is they've modified the surface of the silica gel to put these long fatty acid tails, in particular oleic and stearic acid, which are just, long fatty acid tails. And so if you think about it, what they've done is they've taken a very polar material, silica gel, and now they've modified it to make it non-polar, right? So it's going to stick the non-polar material better than it's going to stick the polar material. That's why we call it reverse phase. And that's typically what's used in HPLC systems when they're doing cannabinoid analysis or just analysis of small molecules. You typically use reverse phase. And then what you do is, so if you think about it like this, that reverse phase is like a big ball of fat that as soon as these cannabinoids get onto that ball of fat, they're going to stick, right? Because they're lipophilic, right? They're hydrophobic, but lipophilic, meaning that they love fat. So then what you do is you change the solvent gradient during the course of the HPLC run. You might start off and it's 100% water. Maybe they usually add a little bit of something like formic acid or some type of acid in there. And usually it's buffered to just make sure that any of the acidic functionality that would be on the cannabinoid acids in particular would be protonated. So they're going to come through in their protonated form. And so what you do is during the run on the column is you have a solvent gradient where slowly you increase the amount of methanol that's running through the column. And now what we know is that if you've ever gotten cannabis sticky onto your fingers, you know that alcohol immediately dissolves all that. So now the alcohol is running through that ball of fat column and is going to start eluting the cannabinoids. So the elution order depends on, a lot of things. Again, if you're looking at cannabinoid acids, because they're more polar, they would typically come out sooner than later. There are also differences, like in how the like you can't, the terpene profiling that Gissy did on GC, you can't do by HPLC, because what you're doing when you're looking at HPLC is you're typically just looking at the cannabinoid profiles. In order to look at the terpene profiles, you really got to do that by gas chromatography. So that's why it usually requires two different instruments to do them. And Ryan, I'm sure you've seen this. I mean, there's methods where you could use gas chromatography to look at cannabinoid profiles, but at that point, you're decarboxylating all your cannabinoid acids, and you're not differentiating the acids from the decarboxylated ones. So I guess, there's benefits to doing both. I guess this quick near IR method is easy in terms of its turnaround time, and it's fairly precise and accurate. I can't really comment on it. I
Well,
don't
it
really have
takes
any.
like 7,000 readings in 55 seconds, and then it generalizes those readings. So it gets it. I'll tell you, we nailed. I don't even know how many. I never heard a person say that was far from a test we've already had, but we nailed a bunch of them, 100 for 100. Like it was like 51% was Remo's dry sift. We had 51% on the Valenveras. I don't know how much it was off really ever. Although people do say they find the terpene numbers to be lower than what they thought they were. But I wonder if that speaks to the unit as much as it speaks to the labs doing the testing for terpenes. Because I did see overnight terpenes go from like 1% to 2%. And now it's like everyone's
Yeah.
got like 3 to 5, 3 to 6% terpenes on their flour. And I'm like, that seems really high.
6 is
Yeah,
really
they're
high.
they're
The
they're
Canadian
they're
labs are.
fleeting. They're fleeting. And the other thing, just as a stickler, I'm a stickler for detail. Nomenclature is important. We really need to be referring to this class of compounds, Ryan, not as terpenes, as terpenoids.
Oh, yeah.
Terpenoids is a much wider class of molecules that include these sulfur compounds and some of the aldehydes and esters as well. So if you could find a C5 fragment that's in those molecules that's derived from isoprene, it by definition is a terpenoid, right? So the methyl butene blah 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 thiol, that is definitely a terpenoid because you could see the isoprene in there. So I think we need to just change our, because terpenes by definition are strictly hydrocarbons. So as soon as you talk about linalool,
Yeah,
that's no
linalool is
longer
not
just a
terpene.
hydrocarbon, right? You
I
have,
know.
yeah, I know.
It just
It's just
depends on what level
it's
you
a
want to.
subtlety.
Well,
Well,
how about this, Dr.
Nomenclatures.
Mark? How about this? You should create a presentation on this and we'll do a presentation with you for an hour one day. I love the idea of doing it, man, honestly, because you kind of got to walk people through and kind of get them to the
Yeah.
point where they can, you can't just jump in like year 4 PhD chemistry work. Like you got to kind of walk us in a little bit. Ryan
Maybe
did a
Yeah,
Mark.
great
well,
job today.
Dude.
again, terpenoids is just a much bigger class of which terpenes are in there, and things like limonene, terpinolene. Yes, those are terpenes. Beta caryophyllene is a terpene, but it's a sesquiterpene, right? It's not a monoterpene. So
No, and that
calling
was
beta
noted.
caryophyllene a terpene,
If
yeah,
you were if
that's
you were paying
technically.
attention, that was noted in the presentation.
So,
So
Mark,
It's just all nomenclature, Ryan. You know that. I mean, I
would you
teach in a
agree?
classroom where I got to get all that terminology correct because those students are hanging on every word I say, right? And so I'm just a stickler for using correct terminology and structure.
Words matter.
Yeah, Ryan, first of all, buddy, I love you. I think you know that. I mean, we've had this bromance for years. I mean, I love that you rely on me kind of like as your chemist up your sleeve. And so, right, there was a post on LinkedIn this week, right? And so, what you want to explain that post to the audience? I mean, what was that all about?
well, someone had they were running, actually, funnily enough, they were running TLC plates, and
Yeah.
they had a molecule that they weren't sure what it was on their plate, and so they were trying to figure it out.
Right. And so I guess the point was is that we were looking at her TLC plate, and what I found is that unlike I guess what you had hypothesized is that
Well, again, Mark,
THC is
I was using reverse plates.
right, right. That's why, yeah, it's super important to get all that kind of detail right because again, these things have very subtle differences. Whether you use alternating current or direct current, that matters too, right? So if you're using one solvent or another, all these things matter. But I guess the important thing is that, yeah, I mean, like there are people who are out there who are using TLC and those methods to profile plants in their garden. And I think it's great to get that message out. And
Yeah, well, if you're
yeah,
just
I,
looking
I
for
enjoyed your presentation today, Ryan.
if you're just trying to tease apart CBD plants, in your garden, or what's a type 1 plant, or they're doing their own breeding, there's no reason to spend hundreds of dollars on a sample to get that through a lab, right? Like a cheap TLC setup, those probably
Yeah,
cost
well,
us
and
three.
also there's the beam test. The beam test has been known for over 100 years, and
Yeah,
that
but
is if you take a little bit of
the beam test won't separate.
ethanolic KOH, right? A
Yeah, but
KOH or
the beam test doesn't separate type 2 and 3 plants though.
no, but that would just basically confirm the presence of CBD.
Right.
So
But
if
if you're using
CBD
those
is
for
present,
breeding, if you're using those for breeding, you want to be able to differentiate between those two classes of plants,
yeah,
right? So, um, beam test for sure it's a cool trick. There's no doubt about it. I used to have that up on my one of my social media profiles for on a cannabis forum, so people could figure out how to do their how to isolate CBD plants. Like I said during that time, because it's so simple. Like it just uses KOH, which is
right.
potassium hydroxide, which for all the growers out there, that's pH up, right? So you use like a couple of drops of pH up in an ethanol extract. Plants that have CBD will leave a little purple residue in the extract,
Yeah.
and it's just a chemical indicator way that you can see if plants have CBD. But it doesn't differentiate between the plants that are pure CBD or have THC as well. So that's why that test is a little bit limiting.
Yeah,
But
but the
yeah, it's about
intensity of the intensity of the chromophora, the intensity of the purple, basically is an indication of how much CBD is there. And you could do on a relatively cheap spectrophotometer a easy calibration curve that just follows a Beer's law
yeah, but you're not going to
type.
do that. Like you're going to use a TLC plate.
I'm
They're
just
too
saying that
much. That's what I'm saying. That's why we
if
use
all
the
you have is a UV spectrometer, you can do a
sure
calibration curve and measure how much
for that
CBD is there.
very small like cohort of people out there watching the show that only have a UV spectrometer. They can do that
Yeah,
for sure. But for people that are wanting to do breeding, you use a TLC plate because it's like $2 a sample versus $150 a sample for a GC, right? And there's just no need to if you're running 100 plants, it costs you $200 versus 10,000 bucks. So but that's one of the things you got to do when you're doing cannabinoid testing or breeding. Sorry, so you got to find a way to get the data for as little as possible. Because sure, like we can go to medicinal genomics and pay like $500 to $800 to have every single plant sequenced, but we're pretty much getting burned in the budget if you do it at that point in time. Um, but for sure, the more money you spend on your breeding project, the more you can learn. Like GCs or cannabinoid runs or like molecular markers, if you can get it, it's all super useful. There's no doubt about it.
yeah. I guess the other thing, Ryan, just to be general, like how much can the way that the plant is actually grown, like if you give the same variety to two different growers in two different environments, they could come out with very different profiles of terpenes. I've seen that myself. I mean, so how much room would you expect to see in terms of variation between grows on a single variety?
You see variation, but like you don't really see like difference. Like for example, if you have like a plant that has like a terpene profile, when we were doing the terpene profiling, one of the things that we'd always do is take a relative terpene profile, right? Because it's like they have you have the chemical signature, but just on a plant that didn't get treated well or was off in the corner of the garden, it just doesn't produce as much volatiles, right? So we would normalize all the terpene data, right, within a sample so that it was really expressed as a percentage. And that made all the terpene profiles look identical.
I mean, I can say, Dr. Mark, that I've seen the same clone that I've grown given out to call it 20 different people, and it's come out 20 different ways. I think that's right.
Outdoors, yeah, I think outdoors it makes a big difference.
Um,
In greenhouse and room to room, I didn't find it was that different.
Yeah,
Like
I would
the
tend
plant
just
smelled different, but the chemical inventory was pretty much the same.
I would tend to think that different varieties can react different to different stresses, right? And so those stresses are going to upregulate certain proteins, downregulate other proteins. Those proteins are all basically a manifestation of the secondary metabolite profile, right? And so
Totally.
it's the I guess just it's a rhetorical question, which is and again, I think it plays to the same notion that we've discussed plenty of times on Hash Church, which is all about terroir and all about the other kind of like secondary aspects of the environment where the plant is grown and how that ultimately affects the expression of the secondary metabolite profile. The other thing I just wanted to mention, Ryan, because I know that we've talked quite a bit about this. So, heat and air will damage some of these most sensitive compounds. You were talking about aldehydes earlier. Aldehydes are notorious for oxidizing up to their carboxylic acids, and they could really, really change. A couple of the esters that you had pointed out there were butyrate esters. Well, I don't know about you, but I've smelled butyric acid. Butyric acid smells like puke. It has the essence of puke. So that's an interesting manifestation where, again, the ester smells like a banana or smells pineapple-y or citrusy and fruity and lemony. But as soon as you hydrolyze that ester, which happens in the body all the time, ester hydrolysis is ubiquitous in biology. Now you have an acid that smells like some putrid molecules. So the C5 acid, so butyric acid and butyraldehyde in particular. And one of the things about butyraldehyde is it's very prone to oxidize up to butyric acid. So any of those butyrates, it's just so interesting that, yeah, what Mother Nature can do. I made this series of esters from this acid. It was a naturally occurring plant derived acid. And the acid really didn't have much of an odor at all, didn't have any fragrance. But man, as soon as I made the esters of those acids, these things smelt like bourbon. I mean, they just had this really powerful, overwhelming fragrance that was not in the acid, but was in the ester. So there is really something about the olfactory response to esters and aldehydes and other carbonyl containing compounds.
Well, I think a lot of that stuff happens too in the hash or in storage, right? Like we always do our analysis fresh off the plant or as close to fresh as possible, right? Like it's like we rush dry it with like, you can use a whole bunch of different desiccants, but essentially you're trying to capture, when you're breeding for the plant, you're trying to capture the plant as it was on the flower, right? There's no doubt that changes can happen post-harvest and in curing. Like this sample here, dude, from what was the guy's name that has the puppy on his shirt, Mark? The terps guy? Waldo's Terps or something like that?
Waldo's terps.
Not terps. He's like a rosin guy, a local rosin guy. I won't say his name on here, but he's got his little puppy on his brand. Anyway, he was at book club this weekend. You weren't there, but
Dan.
yeah, Dan.
Oh, of course. Sorry.
Yeah, and he gave me a sample here because it has this weird scent profile in it. But the more I smell it, it's not from the plant. It's something that's happening in the cure.
Really?
I'm quite positive of it because
I can
So
also
like
smells.
oxidation or something? Some kind of
Well, it's got
oxidation?
that kind of vinegary note to it.
Yeah.
Interesting. Yeah, I mean, I've been, over the years, I mean, I'm sure Marcus, you can test this, like how someone approaches how they freeze dry a batch really affects that translation. And there's real nuance to all this, but it is directly translatable to what you're capturing versus what you're just off-gassing. And there are curves, and every varietal has its own unique, need. And yeah, I think that there's like, in terms of contemporary hash, like there's all these little nuanced steps all the way to that banger. To your point, it's changing composition. And I've experienced this even with fresh press to cold cure. Totally different expressions and sharper expressions from fresh press. A little bit more rounder, but may perhaps, the sulfur compounds are a little bit more prominent because the other esters and all the other ketones and all the other things are changing from, to Dr. Mark's point of view, maybe the aldehydes are oxidizing and changing into different things that we're
Well, and the
not.
vinegar makes sense because a couple of these esters are acetates, right?
Yeah,
And so
totally.
if you
Yep.
hydrolyze an acetate ester,
Yeah.
it's going to smell like acetic acid. Yeah,
Totally.
so that's
So, my point is like I think that there is chemistry happening, and the nuance of it is only natural. I just think it depends on when that consumer or yourself may crack that jar for the first time, and that's your assumption of what that varietal expression is, because that's your introduction to that jar crack is your truth, But if you've seen it all the way through the process from flower to let's say even the first wash all the way down, you're gonna have an intimate knowledge of that transformation. That's just
Absolutely.
neat.
Well, and
yeah. And
you nail it right on the
thiols
head. Like
are
that's exactly
are
what
really
happened.
notorious for oxidizing up. So I was wondering,
Yeah.
like even in like the rosin press, that process, even if you're only using a little bit of heat, you're really not doing that in an air-free environment. And so sulfur plus air plus
Mhm.
heat, you can't keep that shit from reacting. It's going to react.
Yeah, but I will, but I think also the where there's sulfur are what's the scatol? There's scatol usually, right?
Yeah,
So
scatol is
with
not a
that,
sulfur-containing compound. That's an
but
indole, but
right,
yeah,
but
that's the
that
compound
doesn't.
that smells like shit.
Again, that's kind of what I'm talking about. Like that gets heightened because the sulfur goes right, and but that because it's funny. I have this jar that I've been saving. I need to send it to testing. It just smells like complete shit. It's ridiculous, and it's old. It's like 5 years old. It's brown. It looks gnarly, but like the nose is there, and it's really fascinating to me because it should not smell like that. It should smell like nothing. It should smell like, flat.
Send a sample to
Yeah.
me. I'll run it on the GC. I'll tell you what's in there.
Yeah, I'll send the whole jar to you, man. It's all you.
You send a whole jar to me. I might put it in my pipe.
I don't know, man. It might put you to
May
sleep.
not make it to the lab,
Immediately.
right?
Might immediately put you to bed.
Yeah, I mean, Ryan, you didn't say the back end of that story on LinkedIn, which was that, I that's exactly what I said to that woman, right? She was like, "Oh, I don't know what this compound is. Could it be this? Could it be that?" I said, "Well, hey man, send me a sample. I'll tell you what the hell it is." uh, I mean, I love being able to help people out. So if it's little pet research project that someone's looking at and they want to try to figure out what's in their cannabinoids, send me a sample. I'll tell you.
Well, that's a
There you
great
go.
service right there.
Ryan, I was
You heard it
Yeah,
first.
I'll do it just for the goodness of it, Marcus. I'm not like charging people money, but I think like
I wish
the
all labs were like you, Doctor.
yeah,
Yeah.
well, yeah. I mean, I'm lucky because I'm in a college, right? But I think that like
My
probably
buddy
like
told me the other day he spent 40,000 in the last 3 years getting
yeah.
shit tested.
Easy. That's low. I
I know,
mean,
but he's
yeah,
just
yeah,
one guy. He's not
yeah,
even a business.
yeah.
Well, now
Alan
what makes
Varus.
sense? So Marcus, think about that, right? So if you're spending 40,000 and that's the cost of one of those instruments, it's kind of a no-brainer. It's like, hey, maybe I should be, doing this in house, right? Like, how much does that near IR machine cost?
12,800 US.
12,800, man. So, geez.
I mean, made in the US, owned by a Swiss
Now
multi-billion
you still
dollar
have to do
pharmaceutical
so.
company.
This is probably the case in Canada. I know it's the case here in the States, which is you still, in order to be in a dispensary and to have a label and to be in a regulated environment, you still have to do third-party testing, right?
Yeah,
But I think,
that wouldn't
Absolutely.
pass.
yeah,
It's not for replacing your testing
right,
necessarily. It's for creating a better data resolution. It's for
right.
validating your feed
It's
cycle
for
with
doing
macro and
your
micro.
R&D that I think what
Yeah,
Ryan was speaking to is like this is really R&D. These aren't really the results that you'd put on a product in the dispensary, but
No, but it doesn't need to be though, because it's good enough to tell you the difference between the plants,
Right.
right?
Right.
Which is what you're doing. And in all fairness, like, you know, these tests that we're talking about, be it for cannabinoids or for terpenes, they have variability that's allowed within the test. Like for cannabinoids, under the Canadian regulations, it has to be plus or minus 15% accuracy to what's actually in the sample, right? So there's a built-in wiggle room that's allowed in the sample, whereas the terpenes are not regulated, so they don't have to be within 15% plus or minus. And as such, a lot of the
Thank
protocols,
God.
well, it's both ways, because the reality is that the norm is in Canada that they do it to within 30%, not 15%, right? So a sample that is, say, 100 milligrams or 100 micrograms, it could be between 70 or 30 or 70 or 130, which is like half the variation of the sample, right? So it's like that's a lot of variation. And that's probably why, Marcus, you're seeing a lot of these like higher numbers, right? Is because there's they don't have to be that accurate. It's not required in the regulations. So I kind of like the labs, the Canadian labs, for being able to identify what's in a sample, like especially places like High North. But I don't really take the absolute terpene numbers as that actually serious.
Do labs in Canada have a different pricing structure for doing a non-compliance test, like doing like
No,
a quick R&D test?
no,
Like, is that a different price than say getting a
no.
result for on a product for a COA?
No, it's not. So it's really difficult to do. Or if you're not, that's why I'm such an advocate of having the lab in house, right? Because it shouldn't cost you. You don't need to pay all this overinflated cost, right? So for R&D, especially for breeding, where you're trying to analyze plants way early young so you can throw them out, right? You don't want to grow stuff that's not part of your breeding program to the end of at the end of the cycle. It's just in the way.
What does it cost a lot in Canada is the stability tests to get into Europe, which is in some cases $25,000 per
Can be more.
cultivar. Yeah, but
What's that,
then
a 6 month or a 3 month?
yeah, it's probably six, it maybe
6 month
three,
stability test.
and then we were
Is that
also
on flower or on processed
It can no,
product
it could
or
be on like it could be
without.
on vape carts,
Yeah,
like you want to send an oil in a vape cart to Australia, you got to do a stability shelf test. It's got to be, I think, 6 months.
we
Just on
talked
the
to
lot?
wow?
Is that on the lot, guys? Like, or is that you have to prove that and then you're
You
through
just
and
have
you
to
get the
prove that your
signature?
SOPs are stable and that when you send the product, if it's a long time in transition, if it doesn't get to shelf, and like it has to have a certain amount of shelf
Is it
life to it.
is it per batch though, is the question really? Or is that
No,
just
no,
in
it's
you do
general?
it
multiple
once per
batches.
SKU.
Okay.
You do it
Okay.
once
All right.
per SKU, and then
Okay.
could
Man,
you
that was gonna,
imagine?
bro. I mean, I wouldn't put it past me at this stage, brother. Like the things they've already asked us to do to date. It's like,
Well,
what else you got for us?
High North, John Slaughter, or whatever, at High North he's a friend. We talked to him, and he was saying it's not just there are ways you can do cannab, and maybe Ryan, you can step in on this. There are ways you can do cannabinoid profiles where we have like the 20 to 22 cannabinoids, and then you can have multiple varieties within the 20 to 22 rating of THC, for example, and then it doesn't have to be one cultivar was what he explained to us. Like there's ways you can do stability tests for 20 to 22, 24 to 26, and 26 to 28. Does that sound? Yeah, anyway, that's what he explained to us. So there's ways to do different things with stability tests, and it's a really big thing in this new era of international sales. So, especially in Germany.
That's German market.
Well, it makes
But,
sense to do it, to be honest, because I think, Portugal is a place where there's some growth, but largely I think the material is not very good, right? And they need good extracts. And that seems like the best way to do it.
Maybe. I mean, Germany does, I'm pretty sure Germany doesn't allow extracts at the moment. Pretty
Not
sure
yet.
pretty sure Brazil only does extracts, for example, which is yeah, that's odd. But I like it.
Yeah.
But as far as the stability test, I mean that's where the Valen Varus would be really helpful as far as your your feeding schedule and how much because you're trying to produce the same thing multiple cycles in a row. There's one way of doing that, having the exact same environment, the exact same feeding program. And like our greenhouse and our regenerative style, like it probably wouldn't even work in that, to be honest. Like, who knows? Who knows what we're even feeding? You know what I mean? Like we don't feed, so I'd be super curious to see what the Valen Varus would like find. I'm sure we're low on something and high on another. Who knows? I mean, honestly, it's magic out there. But and another thing, Ryan, I had a question. I don't know if what you could tell me about this variety. You gave us some of these beautiful varieties, and I didn't know a lot about this one. And I also wanted to know more about the limoncello, if you could tell me. And then I don't know what Simply Irresistible is, and I forgot about that. So I don't know if you have time, but be cool to know about them.
They're all half sibs. They're all half sibs, so they're part of a larger breeding program. But the Conroy is the Congolese crossed with the sweet skunk. Sweet skunk was the plant itself, right? That I reversed. I made it produce male flowers. It's like a prolific pollen donor, and it's just a haze with a pretty good kicker of osamine in with the terpinolene. Whereas most jacks and the hazes are like I don't know if you saw if you go back, people go back and look at the presentation. The red spike is like so dominant when it shows up, and that's terpinolene. And this is a weird secondary plant in that category that has also a pretty big chunk of osamine. And between that and some of the other volatiles that are in the plant, like probably some of the esters, it has this really interesting way of like the smoke just hangs in the air. Like it's got this kind of odd incensey note that's quite unique to the sweet skunk. And often, like if you light a joint, you can't smell it when you're smoking the joint, but if you leave the room and you walk back in, like it's unmistakable. And
Wow.
it's kind of weed that like if you light a joint at a party, all the real heads that are around the room will be like, you'll see them start looking around like, what the hell is that, right? And so I reversed that plant and I crossed it to a whole bunch of different things, and that's what you got there is a set of plants that were in that. Incense haze is just like it came from someone in New York sent it to me. It was supposed to be the piff. So it's like an incensey kind of haze plant, quite similar to the sweet skunk. And so I crossed the two of them together. That's what the Simply Irresistible is. The limoncello is just the self of the sweet skunk. And then the Conroy is the sweet skunk crossed to the Congolese.
I got to stand next to Ryan while he explained it to John Conroy this week during the Grow Up conference. He told him, "Hey, I named a cultivar after you, John." And John was like, "Oh yeah." It's like the Sweet Skunk Congo cross. Like that is not too shabby.
Well, John is like for people that don't know, John is like one of the super important lawyers in Canada in Canadian cannabis law, and he's really like made a big impact in the. He was he was the head of Normal for a while, Normal Canada. But he's an incredibly smart, accomplished lawyer, and he's done all these like social causes.
He must have been a lawyer for 50 years at this point in time.
Easy, yeah.
So he was my lawyer in 1998 for my Compassion Club case. And that's kind of how I got to know John. And what an amazing thing to stand in court with that man and have him representing you. I was like, dude, this guy's going to sort me out. And then going to Amsterdam to the Cannabis Cup with him afterwards. And like, I can't remember if it was that same year, 98, or if it was 99 or 2000 after the whole case had gone through. But great experiences with John back in the day. So it's nice to see him and his wife still healthy you know, in
His
their
wife's amazing.
late 70s, you know, like still hiking the farm, eating fresh food out of the garden, like just really amazing people. And it's been nice to know them for the last 25, 30 years.
He's similar to Ethan Russo in the sense of just like super kind-hearted person, fun as hell to be around, has a love for organic food and just farming and the garden, you know, aside from the off the charts heady, you know, work that they both do in their different realms. Like, you know, they just have this beautiful personal touch on things. And so I really love John Conroy and his wife. And
he's played a huge role for cannabis in Canada, and is just also one of the nicest guys. But I remember sitting with him in Amsterdam one of these times that Marcus was there. And I don't think you made that dinner, but I went out with a bunch of Canadians in the cannabis scene, and he joined like a little bit late, and so he kind of slid in on the right side of me on this like teppanyaki restaurant, and he was about to do a bunch of cases in front of the Supreme Court of Canada, and so like I sat there and chatted with him about constitutional law all night, which is pretty cool when you get to sit down with one of these like super high accomplished lawyers that's doing like constitutional law about cannabis. It was a pretty cool conversation, and he wanted to know. He's like, you know, he was asking about like my breeding work, and I told him all the sciencey things that I was doing, and you know, we're talking 25 years ago or whatever, and after learning from him about what plants he liked, when the Congolese cross came around, I just thought that was the perfect name for him, you know, to name it that plant after him, because it was, and funnily enough, it turned out that he grew up in the Congo, right? Like he spent a bunch of years there, growing up there when he was a kid. So it's an interesting plant. I'll be interesting to see what you do with those this summer, Josh, because they're really long, right? But if you've got, you know, you've also now got some of those haze seeds, right, that I gave you from my little collection. So you've got you've got a lot of really nice terpinolene equatorials in that group, and the Conroy are quite special too. They've got
that's really cool because we're sort of known for a strain called Zelly's Gift up here in Canada, and we created it in 2014 to be an homage to our friend Zelly, who got in trouble for driving weed across the country in Illinois and got put in jail. So it was super bummer for our family. And so he came out, and I'm like, well, we created this from a Hawaiian God Bud by Super Lemon Haze, and then the Jack Herrera Mother from Eugene, which is well known Jack, you know, from Eugene, is spectacular. And we've just been crossing that every year since. You know, we had the Zelly's Gift, this, that, and the other. It started getting always high in terpinolene, but started getting a very skunky and super sulfury in that sense. So, and it's the kind of, and what I love about the haze style of the terpinolene is it's super rushy, gets you going in the mind, and it lasts all year in bags. Like, you know, when you have a sweet bag, you can't see it anyway. Then you have a sweet bag, and you're an annual farmer, you know. And for this is our
Makes
third.
my heart warm to see to know the bag is there, because I know what that bag feels like. I know you don't have to show it.
And I think we're going on. I think this is our 30th year of growing regenerative style like we do out in our greenhouses and all that. And just having our weed for the year, right? So it's like, aside from like we're a company and all that and stuff, we also are just two people that live in the woods and have a family, and we grow weed and we smoke it throughout the year until we harvest it the next year. So we don't have like weed coming in all the time and all that. So we rely on growing weed in the year and saving it all year, because and if we fuck that up, like our whole family, we're not smoking for the year. So that's not gonna happen. So we put our very much so our focus. And then you can know, like, all I'm saying is like there's bags that 6 months into it smell way better than when we put it in. And a lot of times that haze and terpinolene, it just has that greasy trichome that in the end, it just lasts all year long. And that's what's freaking sweet about it. And that's why I think it's still around. It's still part of the flower game. And when people say, oh, I want flower, you have to throw that in the mix, like the new hash varietals that we all know and love for rosin and the culture club or melt. So many cases, they don't even smell in the flower. It's super weird. They don't smell very much. And then you make it into hash and it's just mind blowing how good it smells. So there is differences in the flower these days. And you got to have different sections for different types of weed. Like I'll grow mostly hazes and maybe this Congo and simply irresistible, all that stuff. Probably in one of the greenhouses that's taller. And just let them be together so they can vibe out together, rise up together, trade unknown compounds by just living together and things like that. So that's just the spirit of things around here, which we love. So we're also, so thank you, Ryan, and we do value this a lot, and we are going to plant skunk man Sam's ashes under these plants, and so that's going to be special for us. We'll probably dedicate one of our greenhouses to like that in fullness, and I think that'd be probably pretty special, I think. And so thank you for setting us up with that, and to continue the legacy, continue the lineage, and potentially add to it
Just
based off your teaching.
just to give you the warning, those ones are female crosses, right? So those are feminized, not the haze, but the other ones are.
You know what? We'll give them the same love as every other plant next to it. I'll walk in there, and I won't even. I'm not going to discriminate.
All
I
plants
believe
matter.
I believe in equal rights and justice. So all is well.
You just won't have to pull any row. You won't have to do any rogaing, put it that way. So
My spacing will be on point. Do you suggest a 6 foot on center? Or for those, maybe a 6 foot grid?
yeah, they'll grow.
Yeah.
They'll definitely grow.
And we don't use the plastic netting either.
No.
I'm not saying you shouldn't, but we just don't. We use bamboo. When it comes to it, we prop it,
Sativas, I
Indoors.
recommend 7 foot, giving an extra foot.
Yeah. They're airier, so they let the air flow through. But yeah, that's going to be an
Top it
interesting
later.
greenhouse next year for sure. So, looking forward to that.
Yeah, hopefully you'll get to walk around that with us. And then I was talking with Drake online here, who's in the chat and everything over in Vernon. And we were just trying to mastermind how we're going to get a Val and Varus machine in our network. And
Dude,
so
hit me up.
I'm going to talk to Kim in Holland. I'm going to talk to Marcus, and we're going to see what we can do, because we're going to make something special out of it. It's not going to just be sitting on our shelf. We're going to bring it out to multiple farms and see what we can do to bring the community up with it. So, yeah.
I was also just with Drake and Katie for the last couple of days. And before that, you mentioned John from High North, and I was with him as well. It was like everyone you've named, I literally spent the last 3 days with. It's a small world. No, not Drake the rapper.
Kind-hearted souls out there, I tell you.
Drake the hasher.
Yeah. Big up,
They're awesome. Shout out to both of Katie and Drake. They helped me out so much at my booth this past week. They were super awesome, and it was really fun having them there.
Give thanks. Nothing but good energy there, for sure.
Yeah, absolutely. And great rosin. We were smoking his rosin. He had nice flavors, just puffing down the convention center like hardcore.
Drake's a good example. I mean, he does Jadam, 100% through and through, from the making of the soil to the nutrients to the foliar sprays and everything, and he probably knows probably more than anyone else about it at this point. He's had amazing teachers, and then the work he's done with the teachers is probably what sets him apart. But I think the Valen Veris machine would be super interesting to put in his indoor grow to see, what the nutrient uptake is, in these plant extracts, constantly going out into nature, verifying, weeds that are beneficial to your, making sure that environment's clean, bringing it in, extracting it, applying it in the right timing. It's pretty special. Pretty special. All natural, nothing from the store. I mean, that's maybe some, some ingredients here and there, but, pretty cool. And it'd be neat to see, what the science came in with it. Along with our farm where we're not using, certain things. It'd be cool to see what the comparisons were.
I
And
mean, it's beyond cool, really. It's important.
I'm
I mean,
super interested in that. I love the grow off that they did in Oregon and a few other spots where they took one clone and gave it out to everyone in the spring, and everyone grew it out and they had a competition that fall. How cool is that? I mean, everyone's got the same plant and they grow it out. And then if you put Val and Varus or different like nutrient uptake stuff in there, I mean, that's the ultimate teaching. You can say, oh, well, this is giving you that, this is giving you this. One's not better than the other. They're just did this one does that and this does this. So choose your own adventure. This would be a fun learning experiment.
Yeah, and it'd be great, for hashers to see everyone's journey from, soil to oil. It really shows, to your point, the choices that you've made, and everyone's got a system, right? And it'd be really interesting to see to take the actual data on that, love to see that happen more. Yeah, I think the other thing we were talking about was the ability to just be able to share more genetics today is really just so amazing because, before it was like these clones were so coveted and we were just when I was given something, it was like, dude, you just can't give it out. And it's just today it's like you can try to do that, but it's just going to get out, right? And I think it's important to share more than it ever has been today as this thing gets larger in terms of industry because genetics are really underserved in terms of consistency right now, to Ryan's point in his presentation, because of you know, sure, we have some companies out there bringing these polyploids and other hybrids that are allowing, this type of, desired consistency, but it's not consistent, to your point, Ryan, as you said before, i.e. the point you made about sterile plants, if you will, that won't be pollinated, right? But I think it's interesting to keep in mind that there's a long way to go, to your point, Ryan, in terms of where we're at and where it needs to go in terms of commercial consistency, i.e. looking at soy as a good example, or another industry like that. So
Why wouldn't you give
You've
away?
grown crops from seed, right, Colin? I mean, you see the differences between growing crops from seed or clone.
yeah,
There's no
yeah.
doubt that crops from seed they bang, right?
No, they do. They really do.
So if we could get to the point where we had the uniformity of clones,
Yeah,
but the ability, and like moms get like HLVD and pests and stuff that they end up spreading around on the crop, right? Like
yeah.
if you could go with seed propagules, it's a different game for cannabis, right? And it's just with the way that everybody's just randomly smashing plants together and not really fixing traits and not stabilizing traits, it just means that you can't really use most of the seeds for production planting,
I
right?
think a lot of the argument becomes more cultural market based. Like, I don't want to spend the time to get there because by the time I get there, everyone's on to the next flavor or whatever next thing, right? And I think that's a tough place to lay your hat because it just takes the idea of discovery out of the mix, and it's almost I guess it's challenges, why we do this, right? It's a hard place to believe that because you're doing all this work and it may end up nowhere, that you should not do it, right? And I think that's a common thing I hear from a lot of very experienced, geneticists that have been making seeds for a long time in other industry too. Because it's like, why spend the time? Well, because we need to, right? And I think if we could get to a place where something is stabilized in a call it the system of ultra organic compounds that you presented today, sweets, call it skunk, OGs. I think we maybe be able to get to that place where we could plant a bunch of seeds and rely on that as like a consistent behavior. Because it's hard. I hear it all the time. Like people are trying to buy seeds, but they're not getting the results.
No,
You
no,
know?
they're not because they're just even like I said. It's just like we showed with the plant breeding kind of Punnett squares. Like you might find that awesome plant, but if you try to cross it to something else, or you try to even just cross it to itself, it's like
Yeah.
the plants that come back are not necessarily the one that went in, right? And so,
Yeah.
yeah, like we either got to go two ways. We either have to really formalize this genetic cutting, this unique genetic individual of a cutting is a cultivar, right? And then you have like a national clean plant distribution platform
Hmm.
that ensures that those plants can go all over the country. And then yeah, then people maybe they have like, the growers that don't want to do the selecting, they can order crops of clones that come clean, HVLD free, like pest free, and you've got like 500 sour diesel clones or something like that. Um,
Yeah. I think
but we
that's the
kind of
dream for
yeah
most, right? Take the propagation out and, go direct to flower, allocate the prop in the veg room into a flower room, and then you're just buying the plantlets or whatever you have
yeah, and that might be
Gen
really
Zero.
difficult. I mean, that's a dream, but it might be really
Yeah.
difficult. The other thing that we can get to eventually is if we power through this breeding and we do all the hard work, inevitably we end up with the same thing but in seed form, right? So that you
Yeah.
you can plant crops from seed and have the chemical consistency that you currently get from clones. It's
Right.
possible, right? It's like all the technologies exist. We just haven't really applied them to cannabis because we're so busy crossing everything to everything, right? So
Well,
it's
that's
like,
my point is saying that because like everyone, I don't know, there's just it's such a I hate hearing that because I'm like, okay, Mr. Debbie Downer, let's talk about let's not talk about what we can't do. Let's talk about what we can do.
yeah,
And
I'm
I
not.
do
Yeah, I don't. I
I
don't
don't
see it
think
as
that's solution based. I think it's like copping out. Like you have all this knowledge that we all wish you had, but you're gonna tell us that it's not worth your time. That's
no, no, no.
basically
I'm
what
not
I
saying
hear.
that. I'm not saying that.
No,
It's
no,
just that we
no,
have
I'm
to
not.
take
I'm
the
not
time.
saying you're saying that. I'm saying that
Yeah.
there are breeders out there that have said that openly, and I think that's wrong because That's why I believe that taking your concepts to some of these breeders that are out there now that are already doing great selection work, they already have a knack for. Obviously, they have a knack for resin. They have a knack for plant structure. Utilizing these concepts only make it's like an athlete. It makes them so much stronger and better. And I'd love to see some of these guys adapt some of these tools because I do think that there's a lot to learn just every day and in the practice of creation of any sort of thing that you do, let alone breeding. So I think that there's already foundational work that this gives like a really, really clear path as to where you can go with it. And
I
it's so
don't.
useful.
I don't see like I feel the same way, but I also think that it's like, hey man, if they want to do the selections and they have the great noses and the eye for architecture, plant architecture,
Yeah.
great. Just be a selector, right? But take the plants that you're selecting and pass them off to a breeder so that a breeder can actually work with them and put them into a seed form rather than just the only thing that anybody can get is like random crosses with that plant and something else.
So that's an interesting concept. So you're saying like, so I hear this in terms of like beat maker and producer, right? Because like in music, there are these like amazing beat makers that pass it to the producer, and the producer does the arrangement and sits with an artist, and they do make the song, right?
Yeah,
And not discounting that the beat maker is not brilliant, but also the producer and the artist can't do what the beat maker does, right?
100%.
Respectfully, right? So
Well, it
that's why
takes,
it
yeah,
works. And I
it
think
takes
in
all
this
kinds.
moment, there could be a relationship there, you
Oh,
know?
I agree. I agree. And like I've had people say to me like, "Oh, you're just trying to take over the seed business." And it's like it's not that at all. It's like if I had technologies to make the seeds for people out of plants that they found, it just frees them up to find more plants, right?
Right.
And then you have this like symbiotic relationship where it's like everybody's, everybody has a role and everybody's getting compensated, right? And you focus on the things that you're doing best. So, yeah, I think that's the one thing that I love about the way that the cannabis community mashes all these plants together. We're trying all the odds. It's like having a million or a billion lottery tickets, right? Like for sure, some of them are going to be winners.
Yeah, no, it's fun. It's actually really fun. Like we were talking on the last episode, we were talking in breeding is where I took an auto, um, and I brought it back to standard, and it just shortened the flowering time in ways that I was looking for it to be 6 weeks. I was trying to get it as low as possible, and it proved to me that you can set goals. Now, there's a bunch of other work that I need to do on it. Like it's too short and squat, right? But you're in the realm to then do more work, and I think
Well,
that
the
that's kind
first
of the
step,
building blocks
the
that I think is interesting that it's really that's where the building blocks I think are important.
the first step is always understanding how the trait is inherited, and the auto condition is a double recessive, right?
Yeah.
Like it's a recessive condition, so you lose it in the first generation cross, but it's actually also co-dominant because it shortens the flowering time. Like one allele shortens the flowering time.
Yeah.
So yeah, like when we were doing it, I helped my friend Roberto, who's now passed away. He developed the line for the CBD crew, and he wanted to have an auto that was CBD. And that's exactly what we did. Is you bounce the CBD status between auto and photo for a couple of generations while you're fixing out the traits, right? Because you can't do it in autos. You can't keep the clones alive,
I
right? So
want
you want
that.
to keep the clones alive to be able to flower them, and then you bounce them back and forth between auto and quicks every couple of generations.
Yeah, but I found the resin and I found, you know what I mean? And I found the flower structure that I was looking for. And I just was like, I got to try to bring this over here.
Yep.
And it's, I don't know, and the flavor profile that we found was just like this really jammy, really deep, gassy jam that was, very berry. It was interesting. It is interesting. Still have a grip of seeds of it. So.
We could really use high CBD autos here on the farm because 90% of the high CBD varieties that we've tried to grow out of Colorado and Oregon, they have a harder time finishing here. It's just, and we've tried 20 varietals over the years, everything from auto in the beginning. Was it Ben Centennial Seeds in Denver? Super interesting. We went out there back in the beginning of the CBD. But anyway, but yeah, here in British Columbia, like it would be super nice to have like some high CBD autos.
Well, it's a single
Yeah,
nuclear trait, Josh. I mean, it's a 1 in 4, right? So with the information that's at the beginning of the presentation, like
Yes,
that's all doable. It's all very easily doable, and you can do it. Like you can take the varieties that you have and really like and introduce the CBD and the auto flowering into them.
yeah, yeah. We worked with the Heart Rock Mountain and we have the amazing auto that we've come out with by combining some Kootenai cultivars as well and stuff, and that's what we have
Well,
here.
we should take that thing and make it CBD.
Let's do it.
Yeah.
That's the plan then. I
It's
want
so
to.
easy. It's so easy. What we'll do is you make seeds of that every year anyway, right?
Yeah.
So what we want to do is time that you have a type 3 plant in the garden when that thing is dropping pollen. And then we're already like on our way to the goal.
And
You
that's
try to
where
get some Harlequin, man, and try to get him some Harlequin to breed into it, for Wade and Wade.
that would be
Wade.
nice because I have some like Harlessu and some of the tsunami, like some of the
Yeah.
stuff. It's like pretty well mixed at this point. It'd be really sweet if there was some kind of Wade or some kind of Harlequin original, to come back and work with would be amazing. So I don't know if that exists.
I'm sure someone listening has some access. I would
I would
imagine.
Yeah,
think
or
so.
you just talk to Monica.
Yeah, I can.
Yeah.
We'll just write her.
Yeah.
We'll just write her.
Yeah.
Yeah. That would be amazing. All right. Well,
Shout out to Monica. I gotta give her a text here in the near future. I was thinking about her the other day.
I wanted to say too, we're like 2 days away from the passing of David's passing, a year anniversary now.
Mhm.
27th, and we're on the 25th today. So I've been thinking about Diana a lot and their family, and just missing Sam and how it's been a year. Crazy.
much love and respect.
I've still got his
Monica-
voice as my ringer on my phone. Howdy, 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 howdy. People are looking at me like, what the fuck is that? I'm like, that is Skunkman Sam.
Monica and Forrest will be joining us in Vienna at the UN.
Beautiful.
We will be hosting our event. Maps is going to be our sponsor, and we're just waiting on the approval from the UN, which we will be getting in the next couple of weeks. And they have their passports. And so we're going to be continuing the legacy and honoring Wade and Harlequin at the international level because people need to know, as well as we
Awesome.
will be making a little bit of history during that as well. But that's a few months away still.
I feel like Puffco wants to sponsor that trip too. I was talking to James the other day and he was trying to figure it out. So you should reach out to him again.
Oh, we have submitted our budget. Yes, they're all good to go. And a shout out for Puffco for willing to put up and actually step up, for the culture, for the real. Because it's not easy to get, volunteers who are changing, their history in their countries and pull them out, and we all go together and kind of go as a cannabis tribe and speak as together. And so it's going to be an interesting, challenging year. Of course, California, first the United States left the WHO this week, and California joined the WHO this week. So, my Veterans Action Council, I'll be wearing my California hat at the UN this year. So,
Awesome, dude.
and a shout out to Diana and to Skunkman Sam on the year of the anniversary. I know Diana went to bed a little bit ago. But shout out to their family and much love.
Absolutely. Miss those guys every day, really. Wade and Sam, they were a special lot. It was a real blessing to have them here for as long as we did. I'm definitely trying to follow the Dr. Seuss way, which is to be happy they were here, not sad that they are gone. So there he is. What's up, Sam?
Super beautiful. And yeah, infinite respect to the man.
Yeah.
Hey, Etienne, I saw the emails go back and forth. Are you guys going to be doing a panel at CanMed this year?
Oh, I don't think so.
No.
No, it did sound like it. They want to charge us to come and to speak, and yeah, it's not really our thing. I let them know how we feel, and we'll see what their response is, but I'm not looking for anything saying yes.
Yeah, that's a pay to play conference.
But look, nothing but love and appreciation for putting us in touch. Not everything always works out, but we
Yeah,
appreciate the effort and the connections.
well, I'm glad to help. Yes, I will be there. Someone's asking in the chat.
You can do your panel and thing right here on Hash Church. Ryan did a wonderful panel this morning. I have a little presentation. I hope to get more of that out of you guys as this year goes by.
I will
Well,
be at CanMed this year. I'm going to be on the regenerative cannabis cultivation panel, along with Dr. Kimberly Gwynn, uh, Dr. Zimma Punja, Matthew Endest, and Dr. Zimmar and Dr. Punya.
Zamir Punja is from Vancouver, right? He's from the UBC, or is it Simon Fraser?
Simon Fraser, it says.
Simon
Yeah,
Uh,
Fraser.
Simon Fraser. Yeah, he's amazing.
yeah,
Definitely. And
he's
we provided him with a whole case, like 12 samples of our herb from our greenhouses, like about 10 years, who knows, it was a while ago. And he analyzed it, and it was right at the beginning of legalization and microbials, and whether you could do even pass microbials outside. And he's a really well respected person in our community here. It's cool that you're going to be on a panel with him. Say something about
yeah,
drag. He might not remember, but that'd be an interesting panel.
yeah, it'll be
Zamir
good.
made just so you know, Josh, he made the first genetically modified cannabis ever. Was Zamir. They couldn't
So
get
there
it
you go.
They couldn't get it to grow back into a plant. So they only made callus, but they did the first step of the transformation. They actually had a really neat little method that they used. When you make genetic transformations, they usually use like antibiotics on the media to kind of select certain plants. And he made this like phosphomanose isomerase gene that essentially like will only let the plants grow that have this introduced ability to break down a certain type of sugar called mannose, so you can use it as a selectable marker and not have to use antibiotics. So it was a good thing that he developed. It was cool. He's a smart guy. He's a really smart guy.
We may be doing a panel up in Canada. We have a group, a barrister, Daphne Elisma, who we work with internationally, wants to bring Michael and I up potentially to have a discussion up in Montreal at a university, potentially in May, but we're still waiting on funding from the government. So, which is, it's its own challenge up there at times. So, but if I do, I'll give you ample time and heads up if I'm coming up that way.
Yeah, absolutely. I'll try to be out in Montreal, and
It'll
I'm
be close
sure
to
Dr.
Mark.
Mark could pop up easy enough. But May, I'll try to make sure I'm at Chimera if you're over there, and come check it out.
I'll come
So
up to
much fun.
Montreal with Dr. Mark. Let's go.
I would like to say that I love you all and appreciate you and respect you. Thank you so much, Ryan, for your presentation this morning. It's definitely marked on our YouTube channel as well, saved on our YouTube channel, and I definitely consider it a huge resource that will be probably looked back on, quite a few times over a good resource, let's call it. So thanks so much, and we're gonna go on a walk, enjoy the mountains and the cold weather. Hopefully you're holding up in the cold over there, Mark and Colin. It sounds pretty exciting. You guys are in the thick of the cold. So
That's
yeah.
awesome. I was just up on Whistler Mountain yesterday. It was cold, brisk, but beautiful. We're definitely not getting anything like what you guys are getting on the east right now, but yeah, I hope that passes through and all of our friends are okay. I know a lot of people who are setting up Jennies and the whole nine yards, getting ready for just being slammed by an ice storm, including my partners in North Carolina. lots of friends along the eastern seaboard there. I did want to shout out my good friend Merlin. For those of you that have watched it, I used to have a show with him, getting cooked with Merlin. He's just an incredible chef, super awesome dude, big puffer. And so before Grow Up this year, he decided that he was going to have a like 20-person dinner, which ended up being 36. And that he would make a 5-course handmade like dinner, Michelin star by him and his sous chef Gabe, for 36 people as just a gift, really. Like he didn't charge a ticket price or anything. So I just wanted to take you guys through a little bit of what he created because it was so incredible. These are presentation boards that he put the first dish on. It was a mushroom dish, and they are dehydrated leeks that has created everyone's logo. You can see my logo's like way over here somewhere, but these are all individual logos. There's High North right there. And he did that through like through a sieve, a kitchen sieve. He dehydrated leeks, ran them through a kitchen sieve, through a template, and created all of our logos. And that was just the first meal, which also was a mushroom meal. It takes about 10 hours to make because there's a process. But he also foraged the chanterelles in the forest himself. So I'm just like, dude, and you can see my bee there. It says Bubble Man underneath. I took a shitty picture. It's a little blurry because there was no light, but this was in his basement with almost 40 people being served by him. That was his girlfriend in the red scarf and another friend of hers who were the two waitresses. They kind of brought everything to everyone. This, I don't even know what this is, but it was some incredible beet dish with the, I can't remember what the red and the little things are. Pumpkin
I don't
seed.
like meats, and that looks delicious.
Dude, it was so good, Etienne. It was ridiculous. So, okay, the first course was a silky mushroom parfait with pickled shallots, fried shiitake, and oyster mushrooms in an edible custom brand stencil made of dried leeks and olives, served with sourdough and gluten-free turmeric tapioca crackers. The pickled beet salad was fresh pea and mustard shoots, whipped basil ricotta, whipped ricotta, and roasted pumpkin seeds. This dude is from another planet. So the third course is a hamachi tuna cruda with ají tigre de leche, cilantro oil, pickled shallots, and sweet something fresh cilantro. It's cut out of my photo there. This was just one of the photos of a couple of the people sitting back. That's Mickey from Rubicon Organics. Sitting next to him is Adam Temple. Then there's Paul, Kevin Wong, a whole bunch of cool people. This was the hamachi tuna meal. It was unbelievable. I could taste it 10 minutes after I ate it. It may have been one of the most delicious things that I've eaten this year. And then he also did a seared filet mignon with potato fondant and red wine reduction sauce. I don't eat red meat, so he gave me just the potato fondant, but he made 35 pieces of filet mignon, and every single piece was cooked and cut to perfection. There's one of Zoom Gardens shot of trichomes. Figured we'd end it off with something spectacular. But shout out to Merlin and Kim and Gabe and everyone else that had something to do with that dinner. Truly one of the most amazing dinners I've ever been to. Easily would have been worth $200 or $300 for the experience and the caliber and quality of food. There was also a 3-way baklava dessert that I didn't photograph, where it was 3 very specific different baklavas, from the bird nest baklava with the seeds inside, the log baklava, and the little round sort of bouquet baklava. Such a good experience. Everyone just chuff and tough while the meal was getting eaten. And it took about 2.5 hours for the 5-course meal. So it was a nice amount of time. Shout out to Merlin and Kim. And maybe I'll have to have them on Hash Church one day, and we'll make something. Maybe one day when we can do a Hash Church, I'd love to get you guys in the same room, at least the regular panelists, the 4 to 5 guys that have been showing up forever. It would be awesome to get us in the same room one of these days. And maybe
Marcus,
when we
were going to do a food episode, right? Like I had a couple people in mind too for that too. So maybe we should do that. Get some chefs on, because man, there's a lot of work going on in that area That's really
Absolutely.
cool.
And Merlin doesn't even really produce a lot of infused. He can, of course, and he has the deep understanding when you do infuse a meal, you infuse the sauces, the icing, of the dessert, not the main things themselves, which allows everyone to still eat the meal without any edible aspect to it. And it also lets you titrate and control your doses when every little spoonful, ideally is a milligram. To me, if you're going to go to a medicated meal, unless you're hardcore and can do 500 milligrams or 250, if you're in that 10 to 20 range, it's probably a good idea for you to be consuming 1 to 2 milligrams. And then you can have, upwards of 10 or 15 servings of that and get yourself up to that 20 to 30.
Well, just in terms of like pairing food with cannabis too, there's like a whole science to that. And actually, there's a lot of chefs now. I know there's this one guy in Denver, and I can't think of his name. I could see his face, his face. He does terpene infusion. I don't think they're cannabis terpenes. I'm not exactly sure, but he's now terpene infusing some of his desserts and pastries, and yeah, really just borrowing from the flavor palette of cannabis is great. It's a great thing.
Absolutely. Tim Powers is asking, how is the cross you described previously, Congo times sweet skunk, um, different from what you guys are talking about? Do you understand what he's asking, Ryan? Are you even there?
I think he's taking a bio break.
I'm here. I'm here.
Did you hear the question?
Something about the Congo sweet skunk. It's the same plant. I think he was wondering if it's the reciprocal cross. It's not. It's sweet skunk pollen with the Congo mother plant.
And did you just cross those, or did you go into the F2 and do the thing you were supposed to do if you wanted to make it
No,
more
that's the
stable?
pure hybrid.
That's the
That's the
pure
pure
hybrid.
hybrid.
Okay.
So Josh, that's why I gave Josh the hybrid and the S1 population so that he can bounce them together. It's kind of the way
nice.
I do it, is make those plants available so that you can take them and work them to whatever line you want, right?
How do you want people to get in touch with you after today's show, Ryan? Do you want
Oh,
to
I
leave?
put up a thing. Maybe I didn't stay on long of it. Let me see.
What are you doing? Putting it into chat?
No, no, no. I'll just put it back up on the screen if it can.
Oh, right,
Weird,
that
that
thing.
one thing
Okay, yeah,
doesn't
I'll
come
write
in.
it into chat and lock it in at the top. Once you put it up, I didn't see it.
Yeah, give me a second. I've got to pull it up again.
Being funny again?
Yeah, it won't, for whatever reason, when I select that one thing, it won't pull it up.
So you got to open up like your whole desktop or something
Exactly.
instead of the specific page that makes it so you don't have to share all your
Exactly.
embarrassing tabs.
I don't know. I have the tabs aren't embarrassing. I just have a lot of them.
I'm just talking shit. I just have a lot of them. Well, I'm supposed to close that up. Oh, right. There we go. There he is. There he is. All right. So if you want to get in touch with Ryan on Instagram, Breeding Cannabis, on Twitter, Chimera Genetics, and over email is capital BRSALES at
Proton.me.
proton.me. The
That's
proton mail. Remember
that's the
Hush mail? Well, that's proton now.
yeah, that's the seed. That's the seed company mail. So if you want seeds, you can email me there or
Yes,
email
that's
there.
specifically for seeds. And if you just want to shoot the shit, I guess you can hit them up on Breeding Cannabis or Chimera Genetics on Instagram and X. Should be
Yeah,
called
IG, anybody can message me, so I think that's probably the best place. Breeding cannabis.
Oh yeah, right. Because I guess on X it's not like that. Hey, you kind of have to get to control a little more as to who's able to do that.
I think there's just much more bots that like troll X, so people like they don't leave it open to everybody. But Instagram, it's pretty easy to filter through them, so
That's
that's
awesome.
probably the best place.
Well, we got 25 more minutes. If there's any things you guys feel we missed, or if there's any questions that anyone has in the audience, Happy to ask them. I did see a couple. One guy asked a question early on about, is a plant a cultivar if it's no longer cultivated? Afghan bull rider as an example. If bull rider is gone, is it a cultivar?
Yeah, so the, what makes it a cultivar is when it's described, right? And the thing that we haven't really talked about is that you might have two plants that are different genetically, but that are, for the described traits, they're the same. So they are the same cultivar. Cultivar is actually what we call a cultivar group. Like train wreck is actually a good example of that because it's just one of those, like, it's what I would call a variety, but it's the same phenotype that keeps coming back. And really, like, Mojave's Zeta plant and train wreck and even like DNA lemon skunk, and I have a plant too that's in the same family. Like, there's just, they all have that jack, even like they're all kind of the same family, right? So I would almost call that a cultivar. But
Mm.
if you start attributing other traits like stem color or some specific bud shape or like a leaf, it's really about, a cultivar is made up by the traits that describe it, right? So it has some unique set of traits that is described and unique about that plant. Like, that's what makes it a cultivar.
Okay. And so if you were to sort of hold out a helping hand to young breeders, young whatever you want to call them, seed makers, they're trying to come up with some varieties. What would be the low-hanging fruit you would pass them above like saying, "Hey, learn all of this stuff that you just spoke about earlier." What would some of the most simplest sort of like, look, if you are going to continue to do what you're doing, at least, maybe do this on top of that and you'll increase. What's the kind of lowest hanging fruit that you could give someone?
The lowest hanging fruit is honestly the stuff that you're already looking at. Like the traits that you deem important in front of a plant, in a plant that you're looking at, they're they're what you should be working with. I think the most important part is learning the techniques that I just showed you today and how to tease apart if a trait is dominant or recessive or codominant, right? And so by applying your own breeding, especially trying to use selfing just to teach you how that trait is inherited, it's like figure out the inheritance patterns of the traits that interest you, and then learn to lock them in, right? So you can do that by self crossing and growing out like even just 10 plants. You can start to understand the pattern of inheritance. So that's what I would do. Um, Otherwise, if you're learning to like just experiment, try making a CBD version of your plant, right? That's a pretty easy one. Try making an auto version of your plant. That's also a pretty easy one. Because they're both recessive traits. Sorry, the CBD isn't. The CBD is codominant. But yeah, and see for yourself how traits are passed around, right? And once you learn that, you'll see that there's like a lot of options, right, on what you can do through breeding.
Well, I mean, look at the whole population of the world. Jesus.
Yeah, yeah, we're all there through breeding. There's no doubt about that.
That is definitely the common strand. Strand. Let's talk about strands, shall we?
Well, you
Oh
can
my
also
God.
see, like, I think this is an interesting thing too, is like, when you think of a strain, the word strain, like, that's an organism that, like, when you breed it, it comes back. And just by selfing plants, or even crossing them to each other, but especially by selfing, you see that the biological type that you see, it doesn't come back, right? It's like, even with the train wreck self, like, only 75% of the plants were repeated the phenotype that was in the parents, right? There was that 25% class that were different. So if it was a strain, they'd all be the same.
Well,
Hey,
what is the?
Ryan,
Oh, go ahead,
I'll
Etienne.
see your Google screen, dude. I think you want to share something or stop sharing because
I
that's
can't
all we're
stop.
doing.
It's funny. I can't stop sharing. It just gives me the option to share. I'm stuck here. There we go.
I
There
can
we go.
stop you.
There we go. Good. Thank you.
There you go.
Thank you, Etienne.
Uh,
It's like on a Google page for like an hour.
Somebody put Strandivars. What's Strandivars?
Yes, Strandivars and strands are a little bit different. Let me explain you the bro science. I learned this on Instagram.
Does that come with the CBDs, plural?
That's what makes the CBDs, actually.
I like the one that some guy hit me with today. He was asking me about a back cross 7 or a seventh back cross generation. And I said to him, well, what's the point of doing that? You're almost getting the same thing. You're getting almost the same thing as you'd get when you did one generation of selfing, right? And he told me that I was an idiot and that it produced something called a PB1, right? Which I guess was like a purebred one. But it's this idea that like, you start off like mixing paint. You mix white paint with black paint, and you get gray paint. And then if you mix it back to the black paint, it becomes almost black. And then you mix it black to black paint again, and it becomes darker black, right? And people think that the same thing happens with back crossing. Like the more times that you cross it back to the mom, that it becomes the mom, right? But that's because they don't understand that you have these two chromosomes that are segregating against each other, and that when you cross to a heterozygote plant or a plant that has two different sets of chromosomes, that you can't ever get to uniformity, right? You can't ever get to stability, and just because of the way that cannabis is, it's highly heterozygous across the chromosomes. So it's like back crossing is not a great. It's not a great method to get to where people think that you're getting.
So that's a good tip of the day right there.
Yeah. Well, that's what all those stupid memes that I've been making are about, right? It's like just trying to hammer home in a funny way or like an obnoxious way that if you just keep back crossing, that's you look around, you look at people's catalogs, and you can see pretty clearly who believes that that's the way to do it. But really, it's like it's a plant breeding step for people that don't know how to breed plants, right? They don't understand the meiosis and the segregation that happens, all that stuff that Mendel showed is reality, right? Which is why it is such a good thing to form as a basis. Is that every time you get a new trait, you're like, okay, how is this thing heritable? Can I rely on Mendel's teachings to try to figure out how the trait is inherited, right? And inevitably, like I know what happens is these people are making a back cross and growing like 5 or 6 seeds, right? And so because they're only growing 5 or 6 seeds, you don't see enough of the phenotypic spread to be able to see that the ratios are playing out in this expected way, right? And when you grow like 100 seeds of a cross, it becomes pretty clear, like, oh look, all these ones are, like the terpinolene dominance is a very unique smell. Like we all know it, right? You could pick it out. You could walk through a greenhouse and go, oh yeah, that one's terpinolene, right? So even just using your nose, like you can separate out the terpinolene ones from other flavor classes. But the reality is most people are only growing, 10, 10 seeds, say, from a cross. So, and if half of those are male, right, you just don't see enough of that phenotypic spread in the 5 females to really understand what the categories of plants that exist within the seed line. So, if you don't grow the numbers, you won't see the ratios, right? It's kind of that simple.
Mm-hmm.
So, and not everybody can, right? Like if you're growing in a tent, you can't cram 100 plants in there. But I see like Mean Green from Mean Gene from Mendocino. He's from Freeborn Selections. He's found this way to keep his plants all very small, and he does, like 8 plants in a little nursery flat tray. So he's, he by keeping the plants incredibly small and just flipping clones or plants very early from seed, he can actually go through and see the smell ratios and the numbers, and keep that into a really condensed size. So there's ways to do it if you're really serious about trying to figure it out. But that's why breeding is a little bit different, is because you're growing plants to be able to see this wide spread of traits. You're not necessarily trying to maximize the weight off of any one given plant, right? So I think that's another reason a lot of people miss it, is because they say, well, I want to grow 10 seeds. I was talking to this one LP last week, and they were like, oh yeah, we do a little bit of in-house screening, and we run 4 seeds Every crop and in every room we have 4 plants that are from seed, and it's like, bro, it's like, how do you expect to find new plants for the market when you're just not growing? Like, this is a numbers game, right? Genetics is a numbers game because it's just about shuffling these sets of gene combinations.
Mm-hmm.
So a lot of the plants, by definition, are not going to be what we're looking for.
So are you saying they're not going to get anything good from those 4 seeds?
It's just much less likely, right? Like, you can hit the odds, right? It's like buying the lotto ticket. Like, you still can hit that one in
Totally.
a.
Somebody still finds a Honus Wagner every once in a while, right?
Yeah, well, yeah. It's not a good reliable way to do it, to be like betting on hope, right? When
Where are they putting their focus when, if you're a cannabis company, what's more important than your cannabis genetics, than your IP?
Bro, I had this conversation with a tissue culture company here in Canada. They were trying to get me to get on board with them. And it's like, guys, if you're outsourcing your IP and just doing tissue culture from other people's plants, it's like you never own anything. You end up being a service provider. And you're splitting the profits with the guys that are giving you the cuttings. It makes the business like barely viable, right? And it's something that's totally missing in this space, because you get most of these people are doing half-ass breeding, and sure, they find a decent plant, but it's like they're not finding anything that's like truly exceptional and unique, or something that is just like bulletproof. Like, that's the other thing that we need. Like, say we don't make these plants much better, but we just make them bulletproof to things like powdery mildew and like higher yields and better plant architecture, right? Better stature, like that. All that shit has a real value for the growers. Like this plant that I put up, the Rolex, they're calling it, and the After Eighth, they're like, those plants save them like 30% on their trim costs in labor, right? Because the flower structure is better. So it almost pays for the royalty just by saved labor cost, right? So there's all these ways that we can maximize plants and make them better for production, if you use like a focused breeding approach. But because everybody's just kind of like swinging and missing, or maybe swinging and connecting and find a plant that has good market appeal, like it doesn't necessarily grow well. Right, and so you end up paying, like, in terms of labor costs, or like, I went to this one facility in where was it, in California, maybe it was Colorado, and they were growing OGs indoors, and they had like 3 layers of net. Like, the cost of the netting and the labor required to like just set that up, it's like, man, if you had a better plant, you wouldn't have to do all that stuff, right? And that like really impacts your cost of goods sold. Right, so like there really is like a good reason to be working with someone in genetics that's really thinking about all these things, and not just doing the Sherblotto thing, right? Because most of the Sherblotto thing is based on hype, right? So.
Absolutely. I just told someone in the chat who said they were paying too much for rosin in North Dakota to get in their car and go to another state. And someone was like, "You can't do that. That's illegal." And I said, "Well, is it illegal if they go visit a friend in another state and that person gets them high with shit from that state? Like, how far are they taking it?"
Well,
We're going down this slippery slope.
Oh, well, no, like honestly, would it be illegal if I went to another state and bought cannabis there? Are you allowed to go to a state and buy cannabis as long as you use it there? You just don't take it back to your state. Is that not legal?
Yeah.
Federally legal, it shouldn't technically cross the state line. But
Right, of course. But if you could still go and enjoy rosin if you lived in whatever state, you could go
Sure.
next door and go hang out for a day or two and get a gram of rosin and smoke it and enjoy it.
You wouldn't be arrested for that if you're of legal age. Nope, that's
You
legal.
just don't bring it back home, right? And then you're good to go.
Correct.
So do that.
Correct.
Now, if he's in Japan, you can't do that, because if you go into another country and you use cannabis in another country as a Japanese passport holder, you're still breaking the law.
Japanese do not mess around with cannabis.
Really? You don't think there's Japanese people hitting dabs in Thailand right
There
now?
are, but that's way more than $94 a gram.
And if they go back and they have THC in the system, they can technically get charged.
Really?
Yeah, they're supposed to maintain their good behavior even when they're out of the country, I guess you could say.
Well, I gotta say, shout out to Japanese people because you always are showing us your good behavior. I love Japanese people. They're so respectful and kind and awesome. And I particularly like Japanese freaks, the ones that are like, smoking DMT and like grown out dreads going on fish tour. I've had some great experiences with some Japanese folks.
Man,
And
the
shout out to,
Japanese growers that I know that come over here and end up working in the grow, they're like also a category of dedication of their own because
it's awesome.
they're making it into like a mastery level course, right into it. So
Yeah, well, a few years ago, we had on the guy from Japan who had to leave Japan because he was promoting medical cannabis, and they were pressuring him so much
That's
regarding
right.
cannabis and how they are, which is interesting to see because they've been drifting actually toward medical cannabis the past few years, again, since Michael and our team changed international law. So, that's where, changing laws matter, because now you can have different countries importing and exporting, and you can have us complain about the quality of certain countries' grows and exports, etc., which, the majority of, Canadian cannabis stays in Canada, right? Whereas Portugal, they export it. So it's interesting the dichotomies that are and economies around cannabis that are growing in the international age, right?
I think we actually export more than we when we distribute in the country too. You'd have to look that up, but we have huge export business right now.
Okay.
Yeah, massive exports like going all over Australia and Germany most, some to Portugal.
Even
Portugal is
Israel still. I met a guy the
Israel
other day
as
from
well. That's
Israel. He
right.
said he imported over 5 tons from Canada so far.
Yeah, yeah, Israel's a big one too. Portugal is kind of like they were using it for what they call GMP washing. So one of the reasons a lot of the bad weeds end up in Portugal is because they're buying stuff. Not saying that all weed from these African countries is bad, but Central American and more tropical places, South American that have legal production in and Africa are washing what they call GMP washing through Portugal. So it's really about like people that need stuff that they're going to make an extract. They want cheaper biomass, and it just has to have gone through like a GMP facility. And it's one of the things that Portugal allows is people to import weed, and when the weed goes through the GMP facility, it's considered GMPized or whatever.
Yeah, that GMP washing is quite something for sure. Definitely a way to get around the GMP, it seemed like for a while. Yeah, we're getting close here. We got about 7 minutes left, and then we'll give it a wrap up. We'll thank the sponsors. I can thank the sponsors now, actually. Shout out to the press club for hooking me up with this cool neon sign. Cool little thing. I did love the one that Alex Ubatuba had at his uh gallery in Denver that said Wook in red. That was also a really good one. Got to get me a Wook sign one of these days. Shout out to the press club for the full support. I'm sorry I didn't get around to doing a giveaway this episode, but I will do one next episode. We'll give away some of their new double bagged bags, seamless bags, and maybe one of their wash spoons. I got to get another one of those wash spoons. I had like 5 of them and I gave them all away. I should have kept one. It's good to pass these things on. Shout out to Puffco for always lacing us up, man. Everywhere I go, I'm hitting these Puffcos. They're so convenient. I can't express to you the addition of worry of carrying a rig over a border. Like, and I'm usually going to Germany or Spain or Holland or wherever, London. But the US is different with my waiver. I just don't want to be traveling with anything. And everywhere I go, I just seem to have, a Puffco and a hot knife and maybe a smaller proxy or a pivot, which allows me to not have to share the unit that I have. I gotta say, I'm really getting big on not sharing, not putting something that goes in my mouth into your mouth after bringing the plague home from Vegas earlier in December. My family and I all agreed it's best if when I'm out in the world, I just don't put things that are in other people's mouths in my mouth. I asked it at the dinner, in fact, as everyone was passing around this huge joint that Mickey had rolled from Rubicon Organics. I said, would you guys ever put your fork in your mouth and pass it around the table while everyone put it in their mouth? They all agreed no, they wouldn't do that. But they did keep passing the joint around and putting that in their mouth, which no problem. I'm not against it myself, but I'm not going to do it. I'm going to make sure I always have another smoking device, vaporizer, in order to not be the guy who's puffing right in front of someone who really wants to have a taste, because I know how that feels. And often I'll sacrifice my pipe and I'll puff the first puff, and then I'll just turn it into a sharing device and I won't hit it anymore. So both of those ways work. Keep me a little healthier. I did grow up. I did not get sick at all. It was great. Smoked a lot with a lot of different people, but just didn't do the sharing thing. Shout out to Bubble Bags, as always, for supporting Hash Church and Grandmaster LED. Didn't mention you earlier, but thank you. He had Mercules, the rapper, at his booth on the Tuesday, I believe, and it was quite a scene. That guy is actually pretty funny, a little bit comical, and quite a musician as well. He takes really mainstream beats and turns them mainstream songs and turns them into these really cool rap songs that seem to come from a lot of he sounds like he's telling the truth when he's talking about pain and trauma and shit that he's been through. So that's resonates with a lot of people. Big shout out to Bingo Lombardi and the whole crew, OG Brick 420 and Wooly Farmer, who was gifted a subscription today by Dread Smokes Weed. Shout out to Dread. Shout out to the BC Trichome Farmer and the Bee Creeper and Drew Robertson and Single Source 76 and Tony Bologna and all the cool folks that are in the chat. You guys make it what it is. 300 plus of you in there all day for 4 hours, listening to Ryan's presentation and listening to the rest of us discuss it afterwards. Really appreciate each and every one of you. I hope to see you next Sunday, where we will be here for Hash Church Season 12, Episode 5. What it'll be, I have no idea. I have about 4 or 5 days to figure it out. I'll probably wait all the way till Sunday, forget to send the links out to everyone on Saturday night, and send them out on Sunday morning, as I did today. Thanks, Dr. Mark and Etienne and Ryan and Caleb and Colin, and everyone else that makes this show what it is. Thanks to me. I'd like to thank myself. Thank you, Bubble Man, for getting the shit done, brother. In the meantime, may the full melt bless your bowl sooner than later. See you next weekend, everybody. Peace out.
Bye, everybody.